there's a new class at the Media Lab, how to AI almost anything, which is happening right now.
Oh, cool.
Yeah.
I think it's time we merged all three of them into a mega
You,
course.
you are actually completely right on. That's something I spoke with Paul Yang. He's the new faculty member that's leading the How to AI Almost Anything. I think that would be the ultimate, right? We digital
Neil,
fabrication, bio, and AI.
Neil and I are starting to work together more closely because we feel like there's a moment of a golden spike, where you put the golden spike between the two railroads,
Yeah.
where his stuff is getting smaller and smaller, and my stuff's getting bigger and bigger. So we might be able to really grow and make anything.
Oh, well, I'd love to hear more about that, George. Yeah, I mean, I think that is uh, where this all needs to go. Hey, Cyrus. Okay, cool. Yeah, Peter, looks like you're in. So just I'll turn it over to you once we get to the right moment. But we'll get started. I'll have some very brief remarks, and then we'll get started with you. All right, everyone, sit tight. I gotta wait for this other classroom to empty out, and then we gotta set up. So it'll be another like five plus minutes, but I'll be back.
Spoken as a true Terminator. I'll be back too.
Recording in progress.
Oh. Nice, nice. Yeah, perfect, perfect. Perfect. Thank you, Michelle. Inger, Peter, can you speak? Just test the audio for a second.
Yeah,
Can you keep talking? Keep speaking?
I can hear you clearly. Not anymore.
Okay. All right, that's good. Great.
Yeah, this is good.
Okay, perfect. All right, everyone. What is that? I think it's just this out. No, you gotta put it. Oh, whoops. No, sorry, I got this. All right. So everyone, actually, I'm going to share my screen really quickly, Peter, first. So just give me 1 second here. If you can stop sharing your screen, I'm going to share my screen. Okay. Now here's a question. If I go full screen, tell me if are you seeing full screen slides that are changing?
Yep.
Yes, we are.
Okay, perfect. Great. All right, so I'm going to leave this here and let me start the recording. All right, everyone. It is that time. I am so, so excited. I hope you all are as excited as I am. It is upon us. How to Grow Most Anything 2025 final presentations. George, great to see you, sir. And so wonderful to see everyone that's joining us online and also in person. Stephen, you have the owl up as okay, great. So, all right, everyone. So very quickly, we're going to launch in because we have a really, really packed day of talks from all of you. But just as a little scheduling, we're going to go pretty much straight through. We may have a chance to take a break, but I apologize. We have a pretty jammed schedule today, so we may actually have to go close to the full 3 hours. We'll do at the very, very end a little wrap up and kind of discussion about future steps, and then we'll take a little group photo together. I think a lot of you have been very busy, so you did not necessarily RSVP for me for dinner, but please do that actually in the context even of this meeting. You guys just send me a message and let me know if you're able to join for dinner. But we will celebrate in person with our TAs and also the students. So please join us for dinner at 6 if you're able to come. Send me a message. And then really important for all the MIT Harvard students, my grades for you all are due Thursday at 5 o'clock. So make sure all your documentation is done Thursday at 9 in the morning. Okay, so that's going to be your deadline. We'll finish getting the grades done that day. And then one other note as well, we're going to send out a survey as a part of this tomorrow probably, or maybe even later tonight. But that'll be a part of your documentation as well. But just stay tuned for that. And then for everybody, today, we're gonna about to see, a couple of hours of presentations from the students, but tomorrow we've got from 9 a.m. until after 7 o'clock Eastern time, 10 plus hours of how to grow presentations from our global students. So please join us for that as well. That is gonna be epic. So really, really cannot wait. But we've got, again, we're starting now what's gonna be an epic marathon over the next 2 days. So really, really excited. Why don't we dive into the presentation? So just as a reminder for everybody, the way that this is going to work, let me share a different screen. So one moment here. So you all can follow along. This was in the email that I sent out as well. But this deck here, this MIT Harvard deck has the order of the presentation. So all of you know what order you're presenting in. And basically, I'm gonna click your Notion link, and then we're gonna drive from here. MIT Harvard presenters, you're gonna sit in this chair. You're gonna drive on with my computer, slide through your Notion page. Up front here, we've got Michelle and Ronan, and they've got two little signs for you. Okay, so we've got a sign that says 1 minute, which they're gonna show when you've got 2 minutes and 1 minute left. All right, so nobody's gonna speak anything. They're just gonna literally put it in your face. So you're gonna know. So you have 6.5 minutes to talk. Okay. After you complete your 6.5 minute talk, you'll have 1.5 minutes where we'll give you some feedback and do questions, and then we're going to move on to the next person. All right. Now, you all know what order you're presenting in. Okay. So get ready when you're going to come on next. Get ready. You can come sit in that chair if you want over there. That can be the on deck chair. And then hop back on, and we'll try to keep the transitions as smooth as possible so that we can kind of move through all the presentations as quickly as we can. So hopefully that is all clear. And with that, why don't we get started? So actually, our first presentation is going to be from Peter. So Peter, if you don't mind sharing your screen now, we'll get going. And since you're remote, I will give you in the chat when you've got 2 minutes and 1 minute left. But with that, everyone,
Sure.
why don't we get started? So everybody, let's welcome Peter. Peter, thank you so much again.
Awesome. Sorry not to be there. I wish I was, but we have a mandatory lab event today. So I'll go. My name is Peter. I'm a fourth year PhD student in Regina Barzo's group, so which is CS group in ECS. I've been personally very much interested in sort of metabolism and the enzyme world for a long time. So this project sort of grew out of a collaboration with a postdoc in Professor George's group, Anush, where we've been trying to develop methods that discover sort of enzymes that do a particular reaction, and the reaction is sort of the synthesis of nucleobased amino acids. So I'll go over just through the order of this Notion page, the high-level overview of why this is important and why we care about these kind of molecules. Has to do with sort of where sort of protein design exists today. So we have the canonical amino acids that we all know and love, 20 of them that we use at all times. The question is, what kind of chemistry does this really allow, and can we expand that kind of chemistry? Specifically, if you can expand it, potentially you can design sort of proteins that have enhanced activity because they have new chemistries. Maybe you can design proteins that have novel chemistries that we didn't have even before. And then the question then is, where do you start? So focusing in on here is, well, you have these amino acids. Maybe we can expand this amino acid library into something else. And specifically, can we expand it such that it has more specific interactions with sort of nucleobases, so the ATCGUs. And so what we have here is a sort of motif that can form a peptide bond like any amino acid. We have a motif that can interact in this particular project, a cytosine, and would have it sort of in like gray in the background. And you can imagine further down the line, you have peptides that have very specific interactions with RNAs, DNAs, or any sort of nucleobase containing molecule. So it could be a GTP, for instance, which a lot of proteins do use. So you have enhanced activity, you have better activity as a result of having new amino acids that are your building blocks. Taking this a step further, you want these proteins to be expressed naturally and organically. Then you need to have an enzyme that you can put into a cell, let's say, that can generate these amino acids that then get charged onto RNAs and then incorporated into proteins that you want. So that's the big vision here. The very specific work that we're doing is the bit in the middle. So we're trying to say, can we find an enzyme that actually would yield this product? Okay, and so we'll go. There's the computational side of things, and then there's the experimental side that we go through. And aim 3 is the big one that we did not get to at the moment. Some just background. The expansion of genetic code, I think, has been motivated for a while. People have done this and taken codons that are redundant and try to put in new sort of amino acids and hijacks the genome in some sense to expand what we're able to do. So this is part of the background that we have. There are also chemical synthesis ways. So you can try to like chemically synthesize molecules that look like this, either by screening a lot, by having sort of chemistry done at high throughput and see what comes out of it. The key question is, how do you make this happen in a cell naturally? And for that, you need an enzyme. So that's where we sort of come in. Right. And so just to give you an overview again, you have the amino acids, you have the kinds of chemical properties that different amino acids have, right? We all learned about like ones that are polar, one of them that non-polar. Some of them are very much active in binding, some of them more structural. And the question is, how much can we expand this list to the specifics of the chemistry that we want? So that's sort of the vision and the impact that we're thinking of. And there are therapeutics that target DNA or RNA because that gets dysregulated in disease. You can have just enhanced activity of all sorts of proteins that use ATP or GTP because you have much better interactions with those GNA nucleobases. So that's really the impact that we have. The innovation of the project is using a computational method and then experimentally doing this in a cell-free system.
You got 2 minutes.
Yep. The bioethical considerations are sort of, the security and data integrity of using these methods, because then God knows who can do what with, enhanced activity. And we want people to still have access to all of this. So I'll just run through the experimental design. One is identification. So thankfully, we have done part of this before where we have 6 sequences that we know produce a similar molecule, not the same one that I showed, but a similar one. So we can take those sequences, fold them with AlphaFold, structurally find similar sequences. So we're under the hypothesis that things that look similar might have similar biotransformations. Some of the code that we had to write for this, we take all of those and then we rank them with the method that we had developed, this ClipZyme method, to get essentially a long list of ranked sequences. That gives you like around 1,000 or so. You pick the top 20 that we would like to go through. So then the CFE reaction, this is sort of the table that's just for a single reaction. What we need is solution A, B. This is from like the New England biolabs. Our substrates, our cofactor, RNase inhibitor, just to make sure that the RNA is not degraded, and the DNA that we care about, those 20 sequences. I have here sort of the plate as we've designed it. So all the sequences 20, some negative controls, positive controls for expression, and then some pooled reactions. And then the protocol for the reaction itself over ice.
You got 30 seconds, Peter.
Master mix prep as well. The protocol is right here. I'll just show some results, right? So these are the AlphaFold predictor structures that we query with. These are all the sequences that you get out. You get sort of 40K. You can filter it down to 4K with e-values. You can sort of align them with the known target that you have. And then the CFE reaction is the DNA construct that we design, the order that we get. Then the actual experiment that we do is, CFE, I think we all know, is relatively straightforward to do. That we did actually end up with a few hiccups. This is what the MS sort of should look like. We didn't get the MS, unfortunately, but in ideal case, this is what they should look like: the negative controls, the positive controls, and then the actual reaction. I want to mention there's just a few things here of the sort of things that didn't have the challenges that did occur. One is, one of the substrates that we wanted to use, the main one, did not dissolve in water. So we had to sort of troubleshoot it in different solutions to make it actually dissolve. And this took a bit of work. The other one is using an electronic pipette that had a dysfunction and sort of ejected some of the solutions onto the plate and other places. So we had to recover some of the solution from the master mix to actually, because we didn't have enough for all 30 reactions. And then we actually also ran out of some solutions A and B that we had to reorder again. So this how all happened in lifetime that I had to deal with as we were going.
Peter, I apologize. We're gonna have to wrap up there.
Yeah,
Yeah. Just give a big round of applause. Thanks to Peter. That was awesome. Let's do. We can do one quick question or feedback comment. I don't know if you've got a question or George. Anybody wants to offer something quickly here? I guess. Okay, Jay. Go, Jay.
AlphaFold's goodness for folding non-natural amino acids.
AlphaFold's goodness in folding non-natural amino acids is Joe's question.
Yeah. So the predictions here are confident. I mean, this doesn't incorporate any non-canonical amino acid. This is like protein that does exist already. In terms of like designing something new and checking whether it binds, like let's say you have a protein you want to change, you want to put in the non-canonical amino acid, make it bind to some biomolecule, like say RNA. How well does that work? It's probably less so. I assume these are still similar enough, like some of the co-folding models that have seen both RNA structures and proteins, because they do co-folding of all. Maybe it's okay because like they've already seen bases, just not in the context of a peptide sequence. But I would assume their confidence does go down just a little bit.
All right, we'll have to leave it there. Peter, thank you so much. And thanks again for joining us remotely. And
course.
let's bring up, yeah, thanks so much again, Peter. And let's bring up Spruce, if you don't mind exiting or stop sharing, Peter. Thank you. And give me 1 second here. Let me remove this spotlight. And then let's share here. Okay. All right. All right, everybody. Welcome, Spruce.
All right, great. So my final project was about cultured meat. I'm going to talk about the argument and the motivation for cultured meat, what synthetic biology can contribute to the project, and my experiment where I tried to engineer slightly more efficient growth media for mammalian muscle cells. The vision here is kind of, I mean, look at our emissions chart. About a quarter of it is due to agriculture. And if you break down what's happening in agriculture, it's mostly things like land use and terraformation. Basically, it's just the animals. Like it's not energy related. Even if we had 100% renewable grid, these emissions would not go away. And it's actually getting worse than you think because meat demand is skyrocketing. Like beef is going to about double by 2050. So, this is going to be a huge chunk of our emissions unless we can solve this. And the goal of cultured meat is to try and get meat that is more energy and resource efficient than just growing the cow. Because if you think about growing the entire cow, we're spending all this land and energy producing this whole animal. We're only interested in the steak. So the goal is, can we get just to the steak using tissue culture practices? The issue with this is it's super expensive. Like if we're going to use off-the-shelf Thermo Fisher parts like we do in this class, it would cost, by my estimation, like $500 to $1,000 for a single chicken nugget. So it needs to be like 100 to 1,000 times cheaper. And that's kind of the project that the culture meat has been on. And this is what synthetic biology can help with. Some of that cost reduction is going to be in industrial processes. A lot of it's going to be in the growth media. So the growth media is consumable. It doesn't get cheaper. So anytime we can make the growth media more efficient or reduce the cost of it, that's incredible. And that's kind of what I was trying to do with this project. One easy intervention here is to add these two proteins, fibroblast growth factor 1 and 2. And this is basically to specialize. Growth media is generic. It targets all sorts of cells. And the aim with this synthetic biology project is to try and like engineer the media in particular to help muscle cells accelerate growth. So the idea basically was to add these growth factors to cell media and see if it increases growth. This has been done before in the literature, not quite with FGF1, I think, just FGF2 and in bovine chicken and salmon cells, here using mouse cells. So no one's done quite what I'm doing, but it's been well characterized. So yeah, aim one here is basically just to create a mammalian tissue cell culture, put in the proteins and measure the cell count. You count this up in basically a grid and find out whether the proteins actually affected the growth. Aim two, hopefully, is to try and drive this all the way through to like a finished meat product. So basically putting together the cells into a scaffold, into like something that you could maybe eat. Looks a bit weird here, but and then finally, like I said, the goal of the entire kind of agenda is to get the cost of growth media down to the point where these things are actually edible products and can be sold at the same price as regular meat. So producing a cell culture is this from literature fairly easy. This part was done for us because we're going to use chicken cells. Eventually, we're not using mouse cells, which are just kind of off the shelf C2C12 mouse skeletal cells. This part was done. Here's my plasmid. Basically, I put in it's not actually the plasmid I used, but basically the same thing, T7. We had two proteins, FGF1, FGF2, with and without this, with his tag and with and without this now E tag that's meant to make it more soluble. In practice, it didn't work, but whatever, I had the other ones. So I could go through the entire protocol. I think in the interest of time, I'm just going to go to our results. and basically just like walk through the wet lab here because we did get something. So yeah, here's the clinical plasmids, the usual heat shock transformation into the E. coli, and then find out that you shocked them too long, try again. And yeah, we ended up with basically 8 proteins on various levels of IgG added, which basically induces the E. coli to start producing the protein. So we had these 8 variants of FGF1 and FGF2. And then the goal is to get them out of the E. coli into a purified form. So we start with basically cell cultures, we lyse them with the sonicator, and then purify them. The purification was really difficult. I think I may have messed this up actually. And then we started with the tissue culture. So this is the part that I really want to talk about. We start with this T25 full of C2, C12 cells. This is what it looks like on the inside. It's about 50% confluent. And we basically broke it up into this 24-well plate. And then we, as I said, basically put in various concentrations of the proteins into the wells and left them for 2 days to simmer. And when we came back, they looked like this. Now, these yellow ones are interesting because that means that the growth media has been used up, which, I hoped was due to like my growth factors being incredible. It's not, it's just contamination. I messed up my procedure. But the left half, which is FGF2, made it. But yeah, this is one of the contaminated cells. It's just a bunch of dead cells. It's ruined. However, and this is the big reveal, the FGF2 wells made it. And if you look at the initial results, this is the control. This is with 4 nanograms per milliliter of FGF2, and this is with 40. And you can see that the 40 has like a considerable bump of cells here. So obviously this, isn't qualitative at all, and there's like plenty more experimentation to do. I didn't actually count the cells because doing this in hemocytomas takes forever, and this was last night and I ran out of time. But qualitatively, these results are pretty promising. And again, only one of these 8 proteins actually worked, but if we had a more robust protocol and did better job with purification, potentially this could be something that really worked. This is by no means like a solution to the cultured meat problem. This maybe got us like twice as efficient media, and we need, as I said, 100 times. So there's plenty more work to do in the field. But I think this is encouraging that, with synthetic biology, we can engineer this protein of this growth media to try and get the results we want. And, in some sense, we're going up against actual biology here. You know what I mean? Cows are not inefficient machines. They grow steak pretty well. But, theoretically, with good engineering, we can do better. A bit quick. I just had to.
No, that's actually perfect. So one question now, though, just to slow down a little bit, can you explain again the mechanism? So you expressed a couple of proteins. What was your strategy again for how to get improved growth media? And like what aspect of the growth media were you trying to optimize?
So yeah, I mentioned up here basically the usual growth media you use in these things is DMEM and a bit of fetal bovine serum. That's another big problem with the growth media, by the way, is that it requires killing a cow to get the serum. But yeah, basically this is just generic, and the idea was to add like relatively small, like I said, 40 nanograms per milliliter concentration of these fibroblastic growth factors, and that should encourage the muscle cells to grow harder, basically, and inhibit the differentiation as well. We didn't have on that test rat. So yeah, basically these are just like the regular cell media with an injection of the proteins that I got out of my E. coli.
Got it. Okay. And so the last little figure, because you're trying to show that relative to with growth factor and without growth factor, and so the control is no growth factor.
Yeah.
And then these two are then your additional protein.
Yeah.
Okay.
As I said, maybe later.
Well,
Could be nothing, but like it's something.
Yeah, well, I mean, again, just it's all, we all know it's a lot to try to get the experimental results to work in the time frame that we have. So it's definitely great to see on the first initial promising result. Any other quick questions or comments? Yeah, Ice, go ahead.
Yeah, very cool. First, one thing I wonder about your maybe like bioethics or maybe like the regulation side. Have you ever checked on like is it easy or extremely difficult to put something from the coli into?
Yeah, as you have surmised, it's really, really difficult to do this kind of thing. I think I hadn't investigated the actual regulatory concerns bringing this to market. Obviously, I don't think there's any cultured meat in the US that use recombinant ingredients on the market right now. It may just be impossible. In any case, it's super difficult, right? Because you're not meant to eat E. coli. And that's a huge bioethical concern, is making sure that these things that we're editing, and especially because, again, to get this down to like cost parity with regular meat, you're going to need to do a ton of like, bioengineering, which is going to introduce a lot of these risks. So yeah, it's going to be a hard path
In Singapore, they were doing a lot of the culture meat because they don't have a lot of lands, so they're trying to cultivate meat research. So you might be able to see what kind of regulation that they might be further away from this.
Yeah, I think chicken actually got approved in Singapore a year or two ago. Yeah.
Spruce, before you go, and for everybody, introduce yourself. Like what degree program are you?
Oh,
in?
I'm a junior. I do course 8, which is physics.
Physics. Okay, we love that. All right, awesome, Spruce. Thanks so much again. Great job. The introductions are nice because it's nice to be like, oh, like I'm a junior in physics and here I am trying to do biology. It's nice to see that. All right, so let's move on to the next. I don't think Joffrey is here. So why don't we move on to Emily next. Emily, I'm going to refresh your page. Yeah, come on up. Is this it? Okay, great. All right, everybody, welcome Emily.
Hey y'all, so I am, okay. Just trying to get Notion to cooperate. I am not a Mac user.
Oh, you just want to two fingers to scroll up and down.
Okay.
Yeah. There you
Oh wait, this is actually not the updated one.
go. Do you want? Does this need to be refreshed or?
We need to refresh on that. But anyway, some background on me. Thanks for listening, y'all. I'm a Master's of City Planning student. So I value epidemiology, which is really trying to understand patterns, especially in public health, and doing a more spatialized understanding of health issues. So that's how I'm trying to integrate synthetic bio with my perspective of planning and design work. Okay, here we are. So
Just two fingers to scroll up and down.
wonderful.
Yeah, there you go.
So just as an overview, the goal of this research has been to understand more of the specifics surrounding Lyme disease. So to also express grow protein that's properly folded, which can be challenged, and focus on improving diagnostics and therapeutics as an overview. So what is Lyme disease? So there are just 2 years ago, there are over 89,000 cases of Lyme disease in the United States of America. And recent estimates have suggested that close to 500,000 people may be diagnosed and treated for Lyme each year. Here we have a nice CDC map of where this is focused. So Lyme disease as a sometimes chronic, but definitely debilitating illness that I myself have had, which is part of my interests. Additionally, I'm also from the exact area where there is the most concentration of these cases, which is New York and the Northeast region overall. So what does Lyme disease look like? You get bit by a tick, a little teeny tick, and something happens. So what's going on here? So I wanted to use this project as a sort of foyer into understanding the protein system. So we see a little guy here and these squiggles. What are these squiggles? I'm so glad you asked. They're what they are what cause these bullseyes, which is sometimes what happens when the spirochetes that are in the tick's blood and it gets exchanged in the tick's gut and it causes these bullseyes. For some people, others aren't quite as lucky for unknown reasons. And so it really depends on when you catch Lyme disease, what stage you progress to symptoms. We see the early phase, it's a lot of fatigue. You have those bullseye rashes that we saw above. It could be even weeks to months after being bit if it is not caught in time and you don't get an antibiotic treatment protocol. Then the bacteria can spread to tissues in the heart, brain, and joints. And late stage, it can present as chronic arthritis. But not a lot of succinct research is known yet about this, and there are a lot of studies that are going on. But the lingering effects are concerning and also very intriguing. Oh my gosh, apologies. Because due to the lingering nature, especially in connective tissue, I was really struck by, all right, what's the current protocol? It's antibiotics, which is typically administered for 2 weeks. And the diagnosis method is a protein purification method that works, but sometimes it tells you you have Lyme disease and you don't. Sometimes it doesn't even catch the disease itself. So, and it's also very subject to interpretation of various labs. And for people who do not get the antibiotic treatment in time because they don't know they have Lyme disease, it does not express, then they can develop even antibiotic resistant symptoms that are unknown at the time to them. So it's quite a terrifying experience to not know why you feel a certain way. So that's just the background. So the reasoning for the ability of Lyme disease to linger in connective tissues is due to spirochetes. These are these ugly little spring-like guys, and they enable evasion of the immune system. Basically, I understand them as they're really good at hiding. They have a spiral shape and a flexible cell wall, which the immune system of human or animal hosts are not used to. So this heavily inspired my protein design and research from understanding these spirochetes have lipoproteins. So basically, they play a critical role in nutrient uptake because Lyme disease cannot create its own energy. It needs a host to survive, and it needs these lipoproteins in order to take in the peptides from the host. And so here is what one of these peptides on the surface looks like. Bunch of squiggles, yeah? Dangerous squiggles, I'd say. So gaps in knowledge around these specific binding peptide systems is not all of them are classified within the protein database. There are at least 2 out of the 5 within the system that are missing. And so that's an overall vision for this project is if we can link GFP with the one of the classified proteins of Lyme disease, OpAIV, and the peptides that correspond with that, that can be a useful detective method for showing where the immune system evasion is occurring and used as a classification tool. So this project highlights like fundamental work for understanding a mechanism of infection and expression. And there's actually been a recent study as of March 2025, which really focused on and helped me focus on where a gap in this knowledge is. So my overall aims are outlined here of trying to design a protein fragment linked with GFP, and then try to, in the future, understand infectivity using different protein purification methods. Hopefully, in the future, being able to use mass spec to highlight and understand more about the crystal structure of these unclassified proteins. So first, to achieve my aim 1, designed a protein based on this diagram. Here is my OpAIV protein that's linked with GFP. This is my methodology of how I did it. Had some assistance putting it into AlphaFold. So this is what the gene fragment I designed would look like. And so next steps is actually getting in the wet lab and doing a restriction digest. This is my methodology for that. There were some unexpected difficulties, which actually made for a great learning experience in the lab. This was me with some other students making the agar solution for the E. coli that I am going to be using to express the gene fragment. And then I performed a restriction digest within the lab, which felt a bit like putting my child into daycare when I was incubating. So that was also a good experience to not hold too much to this experiment. And even though it is an experiment that has involved an illness that has affected me, I also found it very important to maintain that this is a scientific observation. There was some difficulties with the backbones, which the TAs were very helpful in understanding with in attempts to PCR the backbone so I could get the out of a clonal gene so I could get the specific binding sites that I needed. So here's some documentation for that. These are my expected results once I'm able to have the PET28 backbone that responds to my specific restriction enzyme sites. So this is a hopeful dilution of that. And then for aim 2, which is quite doable. Yeah, aim 2, which is quite doable, is just doable, is doing protein purification. Here's some methodology. And thank you so much for your time. And I would love some questions.
Background again, you're in engineering.
City planning.
City planning, okay.
Yeah.
Amazing, I love it. So one question, again, appreciate all the background and the framing and the vision. So your ultimate goal, you mentioned, you made a really compelling case, I think, for the need for early diagnostics for Lyme. So can you say a little bit more about how your vision for that might work? Because my understanding is you figured out one of the key kind of proteins that you want to detect, and then you've got this kind of, GFP couple, fusion protein. So can you say a little bit more about how you want to, what the future of the diagnostic looks like, basically?
Yeah, so the future of the diagnostic, it looks like it's focused within the 3 aims as how I'll define it. So it comes like in vivo expression of the fragment within E. coli, and then understanding from the protein that's linked with GFP, just being able to instant gel electrophoresis, just see what the fragment length is, and then comparing that to the peptide that by a recent study was found to be crucial for the Lyme disease spirochetes, also linking that to GFP and comparing the different protein fragment lengths, and then trying to use that within a second aim of doing protein purification and the Western blot method, which is currently used, but the overarching goal is to try to see if by linking GFP and using these synthetic bio techniques, can we at this research stage possibly improve protein purification diagnostics, and then what are the possible implications of that, and also the implications of that for classifying and understanding more about this transporter system.
All right, we'll leave that there for now.
Thank you.
Thank you so much.
Thank you.
All right, so let's keep it moving along. So up next we have Shrika, and let me should I click this one? Okay, great. Okay, great. Is this looking right?
Yeah.
Okay, all right, everybody. Shrika. I think you may need to just do it from there. Just scroll.
Oh yeah, this is true. Cool. So yeah, my name's Shrika. I study computer science and chem. And so my project was more generally about bioautomation. So I focused on optimizing the ribosome binding site in the GFP protein. And the core part of my project was kind of designing a software system that kind of allows for intelligently orchestrating like lab equipment. So like before we get like into like just a couple numbers, in a current lab, like 8 to 10 hours are probably wasted on manual data handling, and then $10 million are lost annually in productivity from biopharma companies. These are just like a couple numbers that we pulled from the literature. And experimental data, because we have so many different hardware systems, is locked in proprietary formats and really blocks large-scale ML applications because of it. And so because of this, lab automation currently is operating at 30% capacity. And so here are a couple quotes that I've either pulled from Reddit, people I've talked to personally at some of these companies, or Ronan. So my favorite one is babysitting tea-cans is the least favorite part of my job. I spent an hour or 8 watching a robot pipette. This was Evan Holbrook at the Weiss lab. And then I guess like more generally, like the industry theme is that none of the liquid handler companies really care about the ease or use of programming for creating intuitive interfaces that kind of work the way that lab people think. They're designed by computer scientists and hardware engineers, and a lot gets lost in translation. And so with that, my 3 aims were kind of these. So the most simple one that I'll talk about in a bit is creating an intelligent lab automation system that optimizes the GFP expression using iterative ribosome binding site design. And I've integrated the OpenTrons OT2 using a Golden Gate experimental procedure. Aim 2 was a more comprehensive metadata scheme that basically captures the entire design, test, and iterate cycle. So that way you can create this automatic feedback loop that feeds in the data of experiments and automatically generates the next one. And so what this would allow is the knowledge transfer between experiments, allowing for just more smart experimentation. And then lastly, didn't get to this completely, but ideally autonomous closed loop system that is capable of designing, building, and evolving by itself. That also comes in with safety screening mechanisms to make sure that it's not like creating like a bioweapon or anything. With an audit system would be nice. So as like a brief overview, what this kind of looks like is experimental intent, converting that to design, the data portion that I talked about, and then lastly, the data aggregation and analysis. And obviously all this will connect to robotic workflows that are hardware. So the first of these, just as a quick summary, converts experimental intent to design. So the goal here is, could you really just take like stuff from an electronic lab notebook that's kind of a natural language, a paper, maybe you're trying to reproduce a study that's already been done, and turn that into robotic execution. And so my background's in chemistry, so I previously made this for high throughput experimentation plate design, so just like 96 volt plates, and then converting that into a hardware agnostic script generation. And so what I mean by hardware agnostic, right now workflows kind of look like this. You'll have, for example, a Hamilton Venus or a Hamilton star or like a TCAN Evo, and each robot will have its own workflow, like its own software, its own paradigm for how to use it. And the goal is to kind of make this look like this instead. And so create one hardware agnostic platform to control both at the same time. And then lastly, the data aggregation part. A key issue right now is that data is just like messy and like not really tagged with metadata. So being able to automatically parse that and tag that so the scientist knows exactly what was ran and what full transparency into the experiment was key. So for the scope of this class, my high level was to kind of design, like I said, like the 12 different RBS variants and kind of build and test them in a closed loop fashion. And so that kind of took two paths. One was the software, the second was experimentation. And so in regards to software, this is very blurry, but this is a kind of like a demo video of what it looks. So this is kind of like it's very blurry, but this is the dashboard that kind of like shows you what's going on in the lab. Designed a data explorer page like with the metadata tagging. These are all the instruments that's integrated to right now. So we have a couple biotech synergy instruments and then an Agilent instrument. And then I guess like my favorite part is this little, so this is the plate design, and then this little visualization that actually produces the code for the global the Gibson assembly, and then kind of visualizes it in that visualizer over there. And then so for the experimentation, this is the GFP protein. And then if you look at that little blue box on the left, that's the RBS. It's 6 base pairs, and I'm modifying that. And I had a algorithm that I designed that chose out 12 initial RBSs to kind of modify it. And so this is kind of what it picked out. Yeah, and so so this is kind of what it picked out. And then my materials had a little bit trouble getting here, so I wasn't able to run this in the lab, but these are the results that the algorithm predicted. And so I guess this doesn't mean much right now, but variance 1 and then like 1 was predicted to have the highest GFP expression. And what I was planning on measuring in the lab was at a fluorescence intensity of like 509 nanometers, kind of, and like excitation wavelength of 488, just measure the fluorescent units of like basically how bright the protein was glowing in. And so lastly, to kind of wrap up, I guess like I really only finished like 2 of my aims, but like largely it'd be really nice to have this central system that can convert scientific intent to robotic workflow, because it like just industry-wide, it's like very sad how difficult it is to just make use of automation. And then because what that does is produce so much data and so much accessible data, the goal is to kind of leverage that data moat, whether that be generating like proprietary models or like even smaller, just like closed loop automation systems or closed loop automation loops that kind of allow you to automate an experiment instead of what is frequently done right now, especially in chemistry, is something we call like combinatorial search, where you're just like searching through vast design space and it just takes forever. And I'm not sure, I guess, like how much I'm a believer in like the notion of a fully automated cloud lab. I do think humans will have to be in the loop for the next few years, but the next generation CRO, I think, is like looks very much like an automated cloud lab in which a lot of things are automated away. But yeah, so I'll close for now, and we'll take any questions.
Hey, questions from the room. I have a question just to try to understand. So I might have just missed it. I just want to make sure I understand. So, you went through this process of doing this RBS design.
Mm-hmm.
And so for the robotics, can you say a little bit more again about like what actually is part of your robotic loop with this experiment? So because you said the reagent didn't arrive in time, but what parts are automated in this whole process?
Yeah, so typically, if anyone's like used the Hamilton or the TCAN or like the OpenTrans software is arguably one of the better ones, you are like manually like putting in like each step, like I want this tip to do this, right? And so what this basically did is it went from if anyone's used Potato, it kind of, it takes your like experiment, turns it into like a kind of what like the protocols we've been using in LabVAR, and then turns that automatically into code. So you don't have to go through and actually like kind of drag and drop blocks or something. I originally used PyLab Robot to kind of do this, but that was like V0 of it. But that was, I think that's like a whole rabbit hole. But like now it's kind of just integrating directly with the OpenTrons.
But sorry, but the platform, is it a Hamilton or is it an OpenTrons?
It's an OpenTrons.
So the OpenTrons is running and the OpenTrons is doing what? Like what is the
The
OpenTrons?
Gibson assembly.
Oh, okay, so, but then the actual testing of the proteins of the expression is happening off the robot.
Yes, yes. So that was
Okay, okay.
well, I guess it could happen on a robot. If it had like a plate reader attached.
Okay.
But
okay.
no, this part wasn't done because I didn't. Yeah.
Understood. Got it.
Quick question. So actually this sounds perfect. So what is the expected outcome and how would it put that first, like for which obvious variant would be like pattern to expression?
Yeah. So what I kind of designed was like a library of previous RVS. Blanking on the tool name, but there's like this. I think you recommended it to me. That kind of library searches through the previous RVS variants and like use like predominantly common use cases. And so it kind of picked them based on that. And then it was like a Bayesian algorithm. So
All right, we'll leave it there for now. Shrika, thank you so much. Great job. All right. Up next, we have, who is up next? Han Zhang. Come on up. Let me make sure I've got your page correct. Okay. Is this right? Okay, perfect. Okay, everyone, Han Zhang, here you go.
Hi, I'm John. I'm from the Graduate School of Design, and I'm in this interdisciplinary program called Master of Design Studies Ecology, which is equivalent to Sustainability Studies. I have a really long write-up. I'm going to try to skim through it. So the project I'm investigating is fabric-based cell-free biosensor for environmental sensing, and I'm specifically using zinc for testing this project. So I guess the broader vision is really to visualize climate change and like especially for a potentially public engaging platform. That's why I chose Fabric, because it has the potential of embedding in wearables or art installations that could be like a communication tool, and having like something that's more accessible to the public that's outside of the lab. And I'm specifically looking at the freeze-dried cell-free system to like detect environmental signals. And the key influence of this project is actually Dr. Peter Nguyen from the Bees Lab. And I actually get to meet him in person. It was really helpful. So this was like kind of the key paper that really influenced a lot of my designs and the way I structured it. So yeah, so cell-free biosensing has been pretty much studied a lot in the recent years, but most of it is focused on the more like health and diagnostic and specifically sweat biosensors, because sweat contains a lot of the signals that could be useful in practical fields. But I'm thinking about like in a more like everyday setting that could look at the environmental signals. So the vision, so this project is actually not a biosensor. It is a electric sensor that could sort of respond to, I believe, CO2 change in real life. I thought like this kind of vision would be cool if we were to adapt it in a biosensing manner so that we don't need to like use electrics and stuff. And this was a ChatGPT generated image. And yeah, as I kind of talked about, innovation is kind of looking at in a more like design storytelling perspectives. So my first aim is to develop a functional genetic biosensor circuit in a cell-free system, which I did not get there, and I'll get more into that. And the second aim is to test out how the process of freeze drying it with like embedding into the fabric substrate. And this is kind of like a very early stage of like a very early stage of design that I have to like basically to have the biosensor in the little silicon wall that could be embedded onto the fabric. And the third aim is obviously to scale up into either wearables or art installation design, and this is kind of a really speculative like vision that I had of like how it could be displayed. And this is kind of like the recap of what I said just now.
All
Like,
sisters, right?
Yeah.
Give a confession that you haven't told any of your sisters.
Yeah, sorry. Yeah, so the aim one is again to like starting from design, moving on to DNA synthesis and cell free. And then the aim two is to really test the freeze dry technique, especially how it works with the fabric. I had two options actually. One is to like the one I just showed to like have the the second one is actually the one I just showed, is to have them pipette in the whale, in the visual whale, and then embed the whale onto the fabric. And the other one is to like test if it's possible to embed directly onto the fabric. And the aim three is to scale it up. So experimental design, this is kind of the diagram I made for the basic zinc LAC-Z sensor circuit I have. So it's a 3-part circuit. This part is to like using ZNTR to like respond to zinc, and then it will give the signal to the LAC-Z reporter that will provide colorimetric assays. And the reason I chose LAC-Z is because it's more trickier to work with than like GFP, for example, but it has the ability to have a vision in the real life, like instead of like under UV lights. And this is kind of the, I'll skim through it real quick. This is the benchling design. And I initially was really scared that I will fail. So I had a lot of backups. Basically, the first 3-part is to test that how, if there's a leakage in the design, that like it would just turn on anyways. And I have a backup with the same zinc sensor, but with GFP. And then the last one is a pure, an easier form of pH sensor. And this was the orders that we got. And in the lab, oh yeah. So in the lab, there were some unexpected things that happened that's like out of my plan, I guess. The first was that the gene was delivered in multiple fragments. So I would need to conduct the Git assembly, which I thought that I did not need to. And the second is that we don't have the materials for the cell-free kit. So my lab just, instead of testing out the cell-free system, it kind of went into the whole DNA prep and PCR processes. And it kind of failed because there were no visible bands from when I did the gel run. This was the result, and there were basically no obvious visuals on here. And yeah, for I guess future work, first I guess two like short-term directions I think I could work on is take the backup plans that I had, the pH sensor, and test that instead. And also I haven't got a chance to get the nano drop reading for these samples that I have, so I would want to do that as well. And yeah.
Hey, that looks like you had a question.
Oh, I was just wondering if you could speak a little bit too. So when zinc is present, then you turn on expression of ZNTR, which inhibits expression of LAC-Z. And then is LAC-Z a repressor that, or like an inducer? How are you imagining that the LAC-Z interacts with your readout, or is it? I guess what is LAC-Z?
It's I'm blanking on the term, but it has the white, blue, it would like has a color change, basically. And that's kind of what I expected to get out of the outcome.
So when zinc is present, what color do you expect?
So when zinc is present, it would first, react to here, and then throughout the whole sequence, it will turn blue at the end. Yeah, I actually started with with CO2, but I realized that it's just incredibly difficult at this point. That's a really good question. I went a long way to here, and I kind of forgot how did I get here. Yeah, because I was debating on like instead of CO2 one, it's like something that has not like have a specific paper that I could have just copied from, but also something that's more workable with. And zinc specifically, when you talk about like environmental pollution, it has a strong presence in like stormwater and yeah.
Okay, all right, thank you so much. All right, so up next we have Fan. So just give me 1 second to open up your page. Okay. Okay, is this looking correct? Okay, all right then, go ahead. Floor is yours.
Hi, I'm a third-year PhD student in the Department of ECS at MIT, and my project is about degradation of microcystine produced by cyanobacteria. So cyanobacteria is a special type of bacteria that have the capability of photosynthesis, and is a very natural part of the Earth's ecosystem. However, cyanobacteria bloom can cause the production of cyanotoxins, and particularly microcystins, that can cause significant concerns for water safety for both animals and human beings. And this is the structure of microcystine. And current ways to remove microcystine from water sources are expensive and cannot completely remove the microcystins and can produce harmful byproducts. However, researchers have identified another way to degrade the microcystine, which is through bacteria. So a couple of bacteria strains have been identified that can degrade the microcystine through a couple of enzymes. The biodegradation pathway is more environmentally friendly, easy to use, and also more effective in eliminating microcystins. So the pathway, we can see that the ring structure is cleaved, and then the linear structure is further cleaved into smaller molecules through a series of enzymes. And this pathway is encoded by the MLR gene clusters. And for this project, I'm focusing on the MLRA and MLRC genes. I want to genetically engineer an organism that can have this capability of degrading the microcystins in the water sources. And therefore, I was thinking, where should I put it? And so I looked into the cyanobacteria symbionts, because those symbiotic partners, they live in close proximity with cyanobacteria. And therefore, if we engineer those symbionts to have the capability of removing microcystins, then when they live around the cyanobacteria, or if we introduce those symbionts into the water sources, then they can degrade the microcystins in close proximity with the source of production. And so the aim for this project, the first aim is to construct the DNA design and transform the mRNA and C genes into the E. coli with Gibson assembly and conform transformation. After that, the effectiveness of the degradation capability will be analyzed through mass spec. And then the second aim is to improve the stability of the enzymes using computational protein design tools, and then test the efficacy of the designs using the same methods as discussed in aim 1. And then the best design will be introduced into the cyanobacteria symbiotic partner to culture the symbiont. And then the third aim, of course, is to culture the cyanobionts and then introduce them into water sources that have the problem of cyanobacteria blooms. I'm still in the process of N1, and I'll show the results I have now. So this is the DNA design. Because I want to produce two enzymes, and I construct it in a way that they come from the same mRNA transcription. So each enzyme is linked with a fluorescence molecule, and through a protein linker, and this will be a 5-fragment Gibson assembly with a backbone and also with the enzymes and the linkers. The enzymes are ordered from tetst, and then the fluorescence proteins will be PCR from the plasmids available in the lab. And I did the codon optimization and did the Gibson assembly conventionally to get the plasma sequences, primary sequences. And these are conditions for the 5 PCRs with different annealing temperatures for the 5 fragments. And then afterwards, the verification will be performed with gel electrophoresis and nanogel. Expected results are shown here. And afterwards, to Gibson assembly will be performed and transformation will happen. So for the PCRs, I performed a couple rounds of PCRs and gel electrophoresis. The first round, the nanogel showed the high concentration of DNAs. However, on the gels, the backbone didn't show up. The other fragments, 4 of the 5 fragments, these 3 are successfully amplified. And then the second round, I optimized the annealing temperature, but it still didn't work. The third round, so with the help of ICE, we set up this PCR that has customized ingredients and with contents specifically optimized for GC-rich primers. And then I was glad to see that the backbone finally have some bands that are showing up for some different annealing temperatures. The future work is to continue with the PCR and verification, and then go through Gibson assembly. Thank you for listening. Now I will take the questions.
Oh yeah, go ahead, Ren.
So as you process this microsystem or whatever, do you know anything about the smaller molecule products that it's processed into?
Yeah, so I don't know the specific products, but so the last step I show here, like there are actually some other processes that's going down like further that break them down into even smaller molecules that are like harmless. Yeah.
One quick question. I think I might have missed this. So your readout to show that the cyanobionts that it's working. You mentioned mass spec, but what's the main readout that you're looking into?
So like I haven't got to this step, but I'll be using the enzymes to cleave the microcystin and then run the microcystin through mass spec to confirm whether it is cleaved or not.
Okay, so it's mass spec is basically your primary readout.
For the effectiveness.
Yeah, for showing the efficacy of the perk. Yeah, got it. Okay. Okay, great. All right, great job. Good. All right, everyone, let's keep this all going. Up next, we have Esther. Come on up, Esther. All right. The floor is yours.
Hi, my name is Esther, and I'm a first-year student in Master of Design. And my project is about enzyme supplement for the PLA degradation at the industrial compostings. And the broader goal of this project is to make this develop a composite enzyme supplement that supports the PLA degradation in industrial composite system. And in order to do that, I am like trying to immobilize proteins K onto the silica beds via absorption to just make it more thermal stable. So before going deeper into this project, I would like to begin with the introduction of PLA. PLA is a type of bioplastics derived from the starch, renewable sources, and it is known as one of the most promising biodegradable polymer. And because of this positive perception, its production capacity will be like estimated to grow till the 4,605 tons by 2028, which is a lot. And like out of that, 2011 tons will be attributed to the PLA. And not only this one, there's some like regulations and like policy changes around this material. According to the 2019 UN Environmental Report, 27 countries ban on the single-use plastic, and then now they're promoting the use of the bioplastics more. And so knowing that there will be a lot of like huge volumes of the plastic waste in the future, I was really curious about if the existing like the waste management system can like handle this volume of the waste in the future. And in my last like previous research project, I had the opportunity to like talk with the Harvard fabrication team and talk about how those like the real pain points of the worst. And they said that the PLA waste, even if you put in the compost bin, they're often ends up in the landfill and resent to it because the industrial composting facility cannot handle those like increasing volumes. And also according to another survey, it says that PLA is considered as a key concern because it causes the physical contamination and it's hard to compose. It takes around like 2 to 3 months to fully degrade it in those conditions, even if in the composting facility. And the problem with this PLA going to landfill is, as you may already know, that PLA cannot biodegrade in the anaerobic condition, like in the landfill, and it may take around 10 to 10,000 years to be like degraded and release the methane gases during the deposition. And this graph shows that how much like methane gas is released during this degradation. And this is another photo that how PLA is not really compostable in oceans as well. Like you can see, like it doesn't really compose at all. So therefore, this project is aiming to like explore the proteins K, which is the most studied enzyme to break down the PLA and other bioplastic at the lab setting, and explore the potential application of enzyme into the industrial composting system as a solution to those problems. But of course, there's some problem, like limitation with this enzyme. Around like 75 and 80 like Celsius degree, it stops the enzyme activity and ethane nature, which poses a challenge for using the high heat composting environment. So this project aimed to improve the enzyme thermal performance through the stabilization and immobilization, especially using the absorption method. And this method is a physical attachment of the enzyme to bind non-covalently to the solid supports, like silicates and carbones. And I chose it because it's most cost-effective and accessible approach within the time frame and with my knowledge scope. And this is a process, the absorption, like how it goes. And for the vision and impacts, I am visioning to make this some kind of a physical product that looks like an enzyme supplement for the industrial composting facility, and hoping that this can bridge the gap between the PLA's theoretical biodegradability and actual behavior in this composting environments. And also, without changing any like the current infrastructure, I hope that this can be a practical and scalable solution to the urgent problem. And for the bioethical consideration, I think this project definitely raises an important ethical question about the human intervention in the natural waste systems. So the principle of no maleficence must be really carefully applied. So for the project aims, so for the first project, it's comprised of the 4 aims. I'm going to design the HEASTEC, the PET28 DNA plasmid with the proteins K enzyme, and to transform the plasma in the E. coli and express and purify the protein, and to immobilize the expressed protein on the silica beds, and to last but not least compare the contrast, the thermal stability of proteins K before and after the immobilization by measuring the residual activity from the casein digestion assay. And the aim 2 is to actually test the immobilized silica bits to degrade the PLA under the simulated composting condition. And aim 3 is I want to contact this local industrial composting facility in Cambridge and evaluate its practicability. And for the project plan, it consists of the 8 steps: transformation, protein expression, cell lysis, protein purification, and stabilization, immobilization, drying storage, and enzyme residual activity measurements. And unfortunately, I could only get to the protein expression stages. But for the transformation stage, thankful to all the TA who helped me with this plasmid design. We designed the HISTA PT plasmid, the protein is K inserted in the backbone of the T7 promoter, and we ordered it first. And then let me show the photos right away. Sorry, I'm like kicking so much. Okay, diluted with the distilled water, and then ice cube, heat shock it, and ice batter, and then incubate it, and then place on the plate, and it was successfully transformed. Yes. And the next stage was expressing the protein. Unfortunately, there was a minor problem, but I made the starter culture, and then for the expression culture, I forgot to incubate it with the shaking, so the optical density was too low. So I ended up making the second one because I was worried about the contamination in the process of measurements. And due to unexpected circumstances, I incubated for 3 days, which made it very opaque. But I will go back to the stage and make sure that finish the cell lysis and other purification using the nickel affinity chromatography, and stabilizing using this TCI, and I'm not sure how to pronounce it, and Trehoser, which is a common like a solution to make it protein more thermostable. And I think immobilization of silica is pretty much simple, just adding it and stirring it and incubate it. And I'm hoping that somehow I can freeze dry it later. And then the last but not least, I'm gonna like heat up the sample at different temperature and then put the casein substrate and see how the digestion level will be using this UV spectral photometer. And for the challenges, the only thing I'm concerned about is, so since silica gel is not biodegradable, it may not align with the sustainable goal of this project. But for now, with the technology, I think silica bits is the most kind of a sturdy one that I can rely on. But for the future, I will definitely look into the computational tools and see how I can develop it without using that. And I think, thank you. That's it.
Such a great job. Yeah, I'm really proud of you. You had a whole big journey this semester, so it's amazing that you've come this far.
Yes.
So yeah, one quick question. Again, one big part again was around this the immobilization of the protein.
Yeah.
Can you say again why it is that immobilizing the protein onto the silica bead, how does that actually help with the temperature?
Because it makes it more stable, the structure, by like putting into the silica bed.
Okay.
Yeah, and therefore it can endure the it's better, I think. That's what I understood. But
And you're so because you're still you're getting close to getting the protein expressed and purified. Did you mention how you're going to do the immobilization stuff?
oh, I did, but it's pretty much gonna be like steering it and like incubate it. But I'm still actually, to be honest, in a research statement to improve it more if this needs to be. 'Cause
Okay.
It's pretty simple. Yeah.
Okay.
Yeah.
And then after that, you would then try to do some temperature tests. and
Yeah,
see.
yeah.
Okay. Awesome. Great.
This is wonderful. I think one question I actually had was more to your design choices. Like, could you explain a little bit more about plasmid? Love to hear a little more about your thoughts on design.
Since initially I was like going to use different plasma, but there was a problem with like ordering this twist. Like they didn't support that plasma, so I ended up using PT28, and we put this proteinase K into the backbone of the T7 promoter using enzyme excellent or something. Yeah. Yeah, yeah. Yeah, and it's he's tagged to do the purifications. Yeah, I think it's yeah.
Awesome. Should make your official release of them.
Yeah.
All right, we'll leave it there for now, but great job.
Thank you so much.
Okay, and let's keep it going. So it's always lovely, you know, again, I think Esther's is a great story of somebody who's totally brand new to biology and is doing some really great work by the end of the semester. So great job. All right, up next, Ricky. Is this looking good?
Yeah.
All right, Ricky, floor is yours.
All right. Hey everyone, I'm Ricky. I am an MSMBA student at Harvard Business School. So I'm doing business plus a master's in engineering. But before that, I was an ovarian cancer researcher, mostly doing diagnostics. And one thing that is really interesting that I learned there is that in the natural process of ovulation, we actually have this repeated release of reactive oxygen species that are contained within normal follicular fluid. And this fluid is basically like washing over the fimbriated end of the fallopian tube every month, causing DNA damage. And so this accumulated DNA damage as we age ends up sometimes mutating tumor suppressor genes like P53, which is associated with the first step in the carcinogenesis pathway that eventually becomes high-grade serous ovarian carcinoma, which is the form of ovarian cancer that's responsible for 70 to 80% of ovarian cancer-related deaths. So like thinking about strategies for mediating this reactive oxygen species mediated damage has become really interesting. And so when we had Chris Mason come in and give a lecture about expressing the tardigrade damage suppressor protein in from tardigrades in human cells, that was really interesting to me because the DSUP protein, which is short for damage suppressor, actually prevents reactive oxygen species mediated DNA damage. I think mostly due to like changing chromatin accessibility. So my idea was to express the tardigrade DSUP protein in human cells in order to protect them from ROS damage. But rather than just like expressing it constitutively as Chris Mason's work does right now in like HEC293 cells, I wanted to express it in human fallopian tube cells and have that expression triggered by the monthly pre-ovulatory luteinizing hormone surge that precedes this ovulation. So basically, you could time it within the cycle such that you have this DSUP protein ready to like defend the fallopian tube cells right before they're exposed to this follicular fluid. So one way to sort of tie this into the LH signaling pathway, the LH receptor is a GPCR. And so it converts ATP into cyclic AMP, which then downstream activates like a camp responsive element binding protein, which binds to sequences in the DNA that have an upstream camp responsive element. And basically, that's just this short sequence that can be induced by any proteins that are activated by cyclic AMP. So my thought was, if we have this CRE camp responsive element promoter region in right before DSUP, then we could make the DSUP inducible by the LH receptor signaling. So something like one of my important bioethical considerations was that previous work has shown that the DSUP protein can be really effective at preventing DNA damage in some cell types, but one experiment in neurons actually showed that it increased DNA damage and neurotoxicity. So I wanted to make sure that we're testing this really thoroughly so that we know within fallopian tube cells where it hasn't been expressed before, we're having the good effects and not the bad ones. So my first aim, I'll go sort of backwards here, was to express the DSUP protein, as I mentioned, inducible by LH in human HEC293 cells to show that we could use the same system that Chris Mason had already shown, but make it inducible. And then aim 2 was going to be doing this in human fallopian tube epithelial cells, exposing them to some reactive oxygen species rich media like hydrogen peroxide and testing how this is actually protecting the cells from DNA damage. And then finally, the long-term vision is maybe this could be a gene therapy or an mRNA vaccine that allows the human fallopian tube epithelial cells to express the DSEP. Since I wasn't sure that we would be able to do the mammalian cell work, I designed a backup that would express DSEP in E. coli. And since E. coli can't have GPCR receptors, I just used a simple circuit to make it inducible by IPTG. And then a backup to the backup in case we couldn't get the PET28 backbones that I was going to use, which ended up being the case, was just to isolate and purify the DSEP gene from the existing plasmids that Chris Mason has published for HEC293 cells, and prepare those for ligation into custom other backbones. So that's the experimental work I actually got to. I took my plasmid from AdGene, turned it into a liquid culture, purified it using a mini prep kit, and then I did a restriction enzyme digestion. What I did was I cut at EcoR1 and NOT1, which were conveniently flanking the DSEP gene in the Chris Mason plasmid. And then I generated a construct for how I would put that into the PET28A backbone. So I ran my digestion and I got a really nice, so this is sort of the expected like virtual digest. This is the DSEP fragment right here at around 1.3 kb. And so I saw those like really nicely here in the gels. Then I used the gel extraction kit where I actually used like the transilluminator backlight of an e-gel machine and carefully like cut those out, digested them, and purified my DSEP DNA. And so I didn't get great results from that. Like the concentration was super, super low, but I think that's because I was just taking from 2 bands and the gel extraction kit we were using was from over a decade ago. So it might've been a little bit expired. But anyways, I have, DNA designs and protocols for actually inserting the E. coli codon optimized version of it into the PET28A backbone. I have other designs for doing it in human work and then actually doing the induction and measuring on a plate reader how it responds. And I want to briefly mention another thing I thought was interesting was like bioprospecting for the DSEP gene. So I actually collected a bunch of moss and lichen from fallen branches in my own backyard, rehydrated them overnight in distilled water. And then that was me looking for tardigrades. Here's a dead or not quite resuspended one. And so ended up finding a ton of them, putting them on the slide, making my own little tardigrade aquarium. You can check out my videos later. These are all the ones I collected. Yeah, some of them are, they're waking up. Yeah, like their alarm just went off. They're still sort of lazy. And here's a really nice 10x video I took. So this one's sort of, yeah, either dead or asleep with his buddies just hanging out next to him. And so my thought is in the future, since some tardigrades actually have better radioprotective abilities than others, maybe some of the tardigrades we're finding in the wild have much more effective DSEP proteins than the ones that we're seeing and the ones that you can just like order off of Carolina Biologics. So I think it'd be interesting to go out, find these new species of tardigrades, extract the different versions of the DSEP gene, evaluate that, and give like the best version possible when we're transfecting human cells down the line and creating these like cancer preventive medicines.
I think we'll leave it there.
Yeah.
Yeah, just congratulations. Really, really awesome project. And, really, really appreciate all your thoroughness and your thoughtfulness and your kind of ABCD mitigation plans that you were forced to execute upon. But I think, again, really, really appreciate all the great work. Any quick questions or comments for Ricky? I guess one question I have for you is, obviously you prepared a lot here in terms of your experimental approaches. Like, what is your next step? Like, if we give you some little more time, like what are kind of your next immediate experimental goals?
So next step is getting twist orders of the E. coli codon optimized versions of the DSEP gene fragment, incorporating that into the IPTG inducible PET28A backbone, and then expressing that. there are human fallopian tube cell lines that you can get or culture from other researchers. At the Wyss Institute, for example, they have like a fallopian tube on a chip sort of system. And so after doing like co-transfection of my other designs into hex cells, I'd love to get my hands on those cells and actually like put it into human fallopian tube cell lines.
Awesome. Yeah, awesome. Ricky, awesome job. Great job.
Thank you.
Okay. All right. And who doesn't love a tardigrade video? So, all right, let us move next. Sophia. Sophia? Oh, Sophia. Okay, great. Let me open this up for you. I apologize, everyone. I don't think we're gonna have time for a bathroom break, so I hope you all just keep riding with us. You need to sneak out, no worries. Sophia, what are yours? Ooh, and you have samples. Awesome.
Hi everyone, I'm Sophia from Harvard GSD. I'm a first-year Master of Design Studies. And today I'm going to talk about my project. It's the development of an E. coli chitosan biosensor for formaldehyde detection. For the background of this project, formaldehyde is a strong-smelling gas used in various products like building materials and adhesives. It can accumulate indoors from sources like furniture and smoking. Exceeding safe level, high exposure may cause breathing issues and certain cancers. Current detection methods like GCMS are costly and not suitable for continuous monitoring. There's a need for a more affordable and portable sensor for ongoing indoor air quality checks. E. coli and other genetically engineered bacteria can be designed to respond to specific pollutants by expressing measurable signals, for example, fluorescence, when detecting the target compounds like formaldehyde. And this is the structure of ECFRMR, which is a formaldehyde repressor protein that can respond to formaldehyde, which can be used in the biosensor. And this is another example of formaldehyde detection via 3 different types of E. coli growth in biosensors. And at the same time, I'm also testing on the form of biosensors with chitosan, a natural polysaccharide derived from chitin, offers a biodegradable, non-toxic, and film-forming material ideal for biological sensing platforms. Its excellent biocompatibility and film-forming properties make it suitable for immobilizing living cells or enzymes, thus creating stable and passive biosensor platforms. The vision of this project is to revolutionize indoor air quality monitoring by developing a sustainable, low-cost, and passive biosensor integrated directly into a structural material by embedding a genetically engineered E. coli system with a biodegradable chitosan matrix. The sensor will continuously detect formaldehyde. And I think the innovative part about this project is that I want to integrate a biosensor into an installation and having this biodegradable structure as an interface that performs the sensing, and which is a low waste and more environmental friendly approach. I got the idea of the installation from the AquaHydra project from the previous mediated matter group at the Media Lab, which is a 3D printed structure with water-based composites that's made from chitosan, cellulose, and pectin. And in terms of bioethical consideration, I think it's more about the biosafety and environmental impacts of using living microorganisms at like domestic settings, and also the accessibilities of that. For project aims, the aim 1 of my final project is to develop a biosensor that capable of detecting formaldehyde by using engineered E. coli that express formaldehyde responsive GFP and a continuously expressed YFP. The aim 2 is to incorporate the biosensor into a chitosan matrix to create a stable, durable sensing platform and to design an installation or an interface that can house this biosensor within an indoor environment. The third aim is to focus on the user experience and to develop a scalable strategies for industrial manufacturing, aiming to commercialize the biosensor as a practical indoor air quality product. And moving to the experimental design and result, this is a PTR47M4 GFP plasma I ordered from Adgene. which is a biosensor that detects formaldehyde with the FRNR and express GFP when the formaldehyde is present. And this is the bench-link file with more detailed information. And what I did is I had this customized yellow fluorescent protein fragment ordered from Twist, and I wanted to insert this fragment into the existing plasmid so that it will have two different types of fluorescent protein, so that it will always show some sort of fluorescence to see whether the bacteria is alive. And this is the final assembly. I'm cutting at the restriction enzyme is the PLEUT1. Moving to the lab protocol, I first cultured the plasmid from Adgene, and then I mini prep it. And yeah, I have finally got these two samples. With two samples, they have slight difference in terms of concentrations. And I then ran a restriction digest with the plasmid. Like, because we forgot to order the enzyme that I needed, so I just had to find another enzyme that's the closest to the existing to the current location, which is FNAR1. And then I did PCR amplification with the UIFP fragment that I ordered from Adgene, that I ordered from Twist, and then combined with the primers. And I have 4 of these samples I did a spectrum of 4 different annealing temperatures and tested their concentrations. And then I run the Gibson assembly with the fragment and the plasmid and the backbone. And then I run an e-gel. I just find out this morning that I think everything else is kind of visible, but the Gibson assembly is like completely not visible. I think this is when my project started to not work. But I still continue with the transformation into E. coli and then plating. And there's nothing. I just find out like before coming here, and I took Alex plate as a positive control with a BL21 E. coli, and this is mine, which has nothing. I think I'm gonna redo the Gibson assembly. So for future work, about the AIM2, I'm actually half done with that as well, because I in a different design class at the GSD, which I'm doing the design for the installation, which is 3D printed, which we use 3D printed chitosan composites. And this is a prototype. Yeah, so the bottom layer was 3D printed and the top layer was hand extruded because we've been having some like technical limitations. And these are the samples. These 2 are the most successful views I have. And yeah. So what I would do next is to calibrate sensor to the chitosan matrix and to form biofilms that can adapt to the installation modules. And this is a speculation of how it might possibly look like with the fluorescence on the chitosan film. And these are all the techniques I've used. That's all for now. Thank you, guys.
Yeah, so congratulations, a great project. I love again the kind of the synthetic biology thoughtfulness, and then you're combining it with a real kind of design artifacts, and then exploring that interface. It is really great job and really novel in combining those 2 things. Curious questions from folks in the room. I guess one question that sort of follows up a little bit with Michelle's question. So why formaldehyde over other particular tanks to sense?
So my initial idea was to make an air filter, because like chitosan has absorption capacity that like it can absorb formaldehyde, but that involves of like electrospinning and transforming it into a nanomaterial, which is like even harder for me. So I think maybe I can somehow do something related to formaldehyde, but sensing, so that if I have the access of electrospin in the future, I can still do the air filter that can filter out the formaldehyde while keeping the sensing as well.
And then one last question. So, for all of these like biosensors, right, living cell biosensors, like if you want to incorporate it into a building, you have to keep the cells alive. at some point, right? So I don't know if you've thought a little bit about that, like once you actually incorporate with the chitosan, like, how would you
I think that would be like the most difficult part. Definitely I'll need to like test on it. Actually, I think I wouldn't be able to know until I test it out, whether like it's better when it's wet. Or then maybe I'll have a system like that contains like liquid in it, or it can still like have good sensing ability when it's dry biofilm. I need to test that.
Okay, great. Okay, awesome. Great job. And thanks for bringing the samples in. We love being able to interact with those. So up next, I think Joffrey, are you up next?
Yeah.
Okay, come on up, Joffrey.
Oh.
All right, have a seat.
Yeah.
Floor is yours. Go for it.
Hi, my name is Joffrey. I'm a second year on undergrad at MIT in the BE department. And so my project is on basically being able to engineer as pasteuri to make better and potentially smarter biocement. And so I guess, for context, climate change, pretty bad thing. We don't like climate change around here, at least personally. And so, there's a lot of work in a variety of spaces. Basically, wherever you go, there'll probably be some climate change work being done. And one of those places is of course, in biotech. And one of the interesting sort of like fields that like subfields developing within this climate change research is on developing engineered living materials because one of the biggest sensibly one of the biggest contributors to the climate change worldwide and carbon emissions generally is building construction and the most common building construction material is concrete. Like I believe I have it in my head. I believe it's about 8% of global carbon emissions is caused simply by just like making concrete and using it and stuff. So being able to even like kind of explore different alternatives to that, could actually make a big impact on emissions. And so there has been some developing work within biocement employing a lot of organisms. Sort of like one of the most famous organisms kind of in this field is Spores Racina pasteuri, which is a gram-positive alkophilic bacteria that's able to spontaneously precipitate calcium carbonate. And this is notable because calcium carbonate is actually one of the main sort of compounds in limestone, for example. So there's obviously like a lot of potential there. And so within like engineered living materials, there's been a lot of research on the pasteuri specifically, but obviously in biology there are like obviously so many hurdles to like whatever new application you want to do. And so with pasteuri, one of those hurdles is actually being able to actually engineer it, because we want just engineer living materials, not just living materials, because oftentimes the natural capabilities of a certain organism can only take you so far. So there have been some attempts on basically being able to introduce foreign DNA into as pasteuri. There's been almost none in the literature that I've seen in at least published literature, because all of them have failed. And so, for my project on, building smarter biocement, basically the aim one, you said I had like 6 minutes, right?
Yeah. We'll let you know when the 2 minutes are.
Okay. So yeah. Huh?
Yeah, 3 more minutes.
Okay, yeah, and so then my aim one was on producing a transformation protocol for S pasteuri. Notably, it is difficult because it is a because it is a gram-positive bacteria, which means it has a very thick cell wall compared to like E. coli. And so like I used a pretty simple plasmid. I believe it's this one. I believe it was P-veg. And so then I grew S. pasteuride, adopting a protocol from Ren. And so sort of I tried two different methods, sort of the plan A and the plan B. One is making a protoplast, which is essentially digesting a cell wall and then introducing it and then regrowing it and then plating it, and then the other thing was sonopreparation, which is essentially you take the bacteria, put it in solution, put it into like a sonicator, which is essentially an ultrasonic probe, blasted with 20 kilohertz ultrasonic waves, and just let the DNA flow in. So I'll just go through my results in the lab notebook. Here are some materials. This is the original one. This is a sonicator I was going to use. So I used a different one. Here are some samples. This is one I actually use. Shout out to the biomaker space. It is a Branson digital sonifier up to 200 watts. I did it at around 120 watts. Here are some materials. Here are some of like the protoplast transformations, pH testing, PBS. Here's the plasmid P-veg. And so TLDR, basically the protoplast transformation didn't work, as you can see by the OD here. But I actually got something interesting in the sonoperation. And so I was like, okay, let me plate this. And so then I plated like a bunch of it on like a cam plate. I got like a bunch of like biofilm, which is probably because I plated like 700 microliters of it. And so then I decided to plate again, where I plated it on one side from the plate again, and then from the another side from a sample liquid culture. And so I got both of them weird streaky colonies, but they look roughly the same. So either there is another organism that also grows in 30C with the same materials used to grow S. pasteurei that is also resistant to chloramphenicol, or I may have actually done it. That's the
So great set of experiments. And, again, I think we've talked about this a bunch. I think, one of the big challenges obviously is, S. pasteurei as an industrially focused, a lot of people in the industry are focusing on this enzyme. So the sonoperation, so had anybody else tried to do sonoperation before that you've seen with S. pasteurei or?
With S. pasteurei specifically, no. There have been a few papers on it being done on like E. coli and this other sort of like very kind of hardy organism as well that both work. So this is probably, so I think this is like the first time it was tried specifically on S. pasteurei, I believe.
But have you seen it for other kind of gram-positive?
Yes. It was used for, I believe, something like Thermobacter, kind of one of the very like thermophilic, like, organisms. In nature, it was like used for that. Instead of a probe, it was an ultrasonic bath, and that also led to transformation. But there was another paper on E. coli, which is negative, which used a straight up probe, and that also worked. So for this one, I used S. pasteurei with an ultrasonic probe because that's just what the lab had, and like I hope this is made of wood. Knock on wood, it worked. Or there's again some other organism grows at 30C with the same requirements that is also resistant to CAM for some reason that grew on the plate.
So well, definitely. I mean, hopefully you can sequence it or do some other tests and verify for us. But yeah, keep us posted on how that goes. But Joffrey, yeah, great job.
Great job.
All right. Let us keep it going. So up next, we've got Yolanda. Open up this page here. Okay, great. And Cool. A couple of cancer-related projects. All right, Yolanda, go ahead. The floor is yours.
Hi, everyone. My name is Yolanda, and I'm a first-year undergrad at MIT, majoring in CS and molecular biology. And my project is de-immortalizing ovarian cancer using RNA interference. To start off with, ovarian cancer is one of the deadliest cancers for women, particularly because it's often diagnosed at a late stage and resistant to many standard treatments. So I wanted to explore a little bit of a different strategy for combating this condition, because one of the hallmarks of ovarian cancer is increased telomerase activation. And so normally when cells divide, part of the DNA at the end of chromosomes gets lost. This is the telomere region, and telomerase is an enzyme that extends the telomeres, as you can see in the figure, which enables cells to essentially divide forever. And you see this in 80% of ovarian tumors. So for my project, I want to employ small interfering RNAs to selectively target and knock down the telomerase gene, specifically the catalytic subunit, which I'll refer to as hTERT. And as you can see in this diagram, by selectively silencing this gene, we can hope to increase senescence of cancer cells and basically suppress their increased proliferative capacity. So I was really interested in this project's concept because I currently work as a UROP in the Belcher lab at the Koch Institute, and we do a lot of ovarian cancer work. So I actually wanted to give them a shout out for helping me out with this project in providing the SKOV3 ovarian cancer cell line for my experiments. And for my project aims, number one, I hope to transfect human ovarian cancer cells with hTERT targeting siRNA. And I'll talk more about how the mechanism of how siRNA works later on in my section on designing a custom siRNA. But essentially, this should cause decreased telomerase expression, which we can measure using a qPCR assay. Further down the line, I would hope to evaluate some downstream effects of this, such as doing functional enzyme assays or evaluating telomere length, or the rate of cancer cell proliferation. And my long-term vision is that I think there's a lot of potential for RNA-based therapeutics, particularly targeting hTERT, because this is expressed, like I said, in over 80% of ovarian cancers, but expressed at very low levels or not at all in normal ovarian tissues. And so this should hopefully limit off-target effects. I think there's also a lot of potential for packaging siRNAs with nanoparticles, since cancer cells tend to uptake those at a higher rate, and this would solve many of the limitations like low biological stability of siRNAs. So that's the long-term vision. And for that, it's also really important to consider things like ensuring non-malfeasance. There's many, many rounds of testing that has to go into designing any potential therapeutic. First, starting off with in vitro work, just using cells in culture. And then if that seems promising, move into mouse studies, then further on with all the phases of clinical trials. So feel free to read more about this in my documentation. But I wanted to move on to some of the experimental protocol. So here are the materials I ordered from Thermo Fisher with a little bit of a justification on why I chose these specific assays. I wanted to focus on the most direct way to measure my results, which is quantifying telomerase expression directly, reverse translating mRNA into cDNA, and then running qPCR. So how exactly do siRNAs work? It really takes advantage of the cell's like native endogenous machinery for suppressing certain genes. siRNAs, as you can see in the graph over here, are short double-stranded RNA sequences, and they are recognized by the RNA-induced silencing complex, which binds to the guide strand of the siRNA. And then that guide strand is complementary to the target mRNA sequence, so they can bind, and then the Argonaute enzyme, which is part of the complex, will then cleave that mRNA. So I took the hTERT mRNA sequence, and following some of the guidelines for designing siRNAs from the literature, I found adenine dinucleotide sequences selected a few target sequences based on their GC content and some other criteria. You can see those target sequences here. I also ran BLAST on these to make sure they didn't have any homology with other known sequences in the human genome, and then selected the two that I wanted to make from Thermo Fisher. After that, I generated the sense and anti-sense sequences for those siRNAs. And I also purchased a positive control and negative control, which are siRNAs that are known to work on telomerase and one that doesn't work on any sequence in the human genome. And now I can move to the experimental protocol. So I obtained SKOV3 ovarian cancer cells from the lab and incubated to 50% confluency. You can see here on my 24-well plate, I've labeled all of the experimental conditions, the different controls, and my 2 experimental siRNAs. Here's the transfection protocol. And then I used TACMan qPCR. Or my plan was to use TACMan qPCR gene expression assays, which have fluorescent reporter molecules that will only fluoresce after DNA telomerase encounters the probe during replication. And then the assay measures the total amount of fluorescence to see how much of your target sequence is present. So more protocol on doing the PCR. And unfortunately, my reagents weren't delivered in time. So instead, for my results section, I've kind of given an idea of what I expect to happen. So the output are a series of amplification curves, as you can see here. And for each of the experimental conditions, the ones where there's no transfection or it's a negative control, I expect to see a low CT value. And that means the CT is the threshold, is the cycle number where it passes a certain fluorescence threshold. So the lower the CT, the more target DNA was present in the sample. And for the siRNAs that work, CT should be higher, showing that there is less target telomerase DNA in the sample at the start. So you can see this is what I would expect to happen. And then there are some equations I can use for calculating like how much hTERT is downregulated after I introduce the siRNAs that hopefully work. So I'd love to carry this out if the reagents ever come. And yeah, thanks so much.
Yeah, really, really great job. And again, I appreciate also the work with the, through the developer lab, being able to work with some real primary cells. Can you talk about, so if you're successful, can you just say a little bit more about how you're imagining this actually working in a clinical setting?
Yeah, so the siRNAs would produce a pretty transient effect, lasting like 3 to 5 days, and then just decreasing telomerase expression. So actually, I think something that might be more valuable is trying out like a different form of RNA interference, like shRNAs, which would have a longer lasting effect that decreases cancer cell proliferative capacity. And then I think it would be valuable to try combining this with other therapies, like just like a chemotherapeutic drug, and seeing if there's potentially a synergistic effect, where the effect of both of them together is more powerful than any of them apart.
That's great. That's great. Okay, Yolanda, great work. Congratulations, Dan. And yeah, hopefully your materials should be writing soon. So I hope you get a chance to try that in the lab. We'd love to see how that goes. All right. So up next we have Alex. Alex, come on up. Everybody, I hope you all are all doing okay. We got one more hour to go. All right, Alex, the floor is yours.
Hello, everyone. My name is Alex. I'm a first-year SMARCS AD student. That means I'm in the Department of Architecture, and I'm doing my master's degree in MIT. So the title of my project is called Nature Glows for Environmental Justice, and we will have a tour before we see like my experimental protocol and like my protein design and my what I'll do in the lab. So first of all, like my inspiration and my background was a paper that I read last semester here, and it was about some communities in Lake Michigan, which they could not actually identify why some of their livestock were dead and why some of the people of these communities were becoming sick. So after further research, I found that these areas have been polluted with PFAS, and it's like leading serious health issues for both residents of these communities and like in the whole US in general. At first I was trying also to understand what is PFAS. So here I provide like the two most common PFAS that we can identify in water here in the United States. So PFAS are called forever chemicals because they have a very strong um, a very strong bond, which is called carbon fluorine bond that repel oil, water, and stains, and can also be very heat resistant. And PFAS are actually very broadly used in everything that can repel water, everything that can resist to high heat temperatures. And like some of these are like some evidence that I have that like how broad is like the uses of PFAS in our everyday like items. So the danger of PFAS is that when it accumulates to our bodies, it's actually most possible for us to have cancer at some point and to make our immune system inefficient. These are some data that I found also in Lake Michigan, that all of the fish that were actually all the fish that they tested there, they all had a huge concentration of PFAS. And then this is a map of the United States showing at the same time like that the PFAS concentration in like all these areas is imminent. And then some other also data that I have based on the PFAS concentration. So about my vision. So since I'm an architect and like I'm very interested in building environment, I would like to have like a biosensor or something that could tell us if our environment around us is polluted by PFAS. So I was trying to find a way if I can develop a biosensor and then embed this biosensor in a building material, and which I decided to be like a brick based on a mycelium brick. So my vision, but my broader vision is like to give local communities the ability to detect PFAS in their environment on their own, simply by observing their built surroundings. There are also some bioethical considerations which are more political. I would say that I think that if this project will be developed, I think we'll have a riot here. But okay, when we go to implementation stage, so a key resource of inspiration of my project was an experiment that shows how PFO, which is like one of the two PFAS that we saw before, how this can be like identified in by in E. coli how this I will start again. So yeah, this paper actually talks about how PFAS can be identified. Yeah, that's all. Full stop. And then so I was also trying. So my first step was for me to try to understand how I can actually here I have a diagram. So yeah, I think this is how this biosensor works. So actually my backbone is like the PET28A, and we engineer the plasmid in order to have GFP and also another protein which binds with PFAS, and it exists in the human liver. It's a human liver protein. So actually the plasmid expresses the express the protein in E. coli, and then when this protein actually identifies or is connected to PFAS, actually it changes the structure of the protein, and like changing the structure, that means that the fluorescence of this protein is increased. So there are some data also that there's a bit of fluorescence before the protein connects to the PFAS component, but also there are also other literature that supports that when it binds to PFAS, it's more the data are more are higher. So my first aim for this for the project is at first to design the DNA plasmid to express the protein, and then express the biosensor into a protein, the biosensor protein into E. coli, and then test the biosensor by comparing its response in the presence of PFAS. My second aim was to do the same exact experiment, but instead of using GFP to substitute it with chromoprotein, because I wanted my final result to be identified by naked eye. And then the aim three of my, and the broader vision of my project is like, if I can embed this biosensor into my mycelium-based rig.
Got like 30 more seconds.
30 more seconds? Okay. So this is my plasmid design that we did here. And then what is interesting to show you? Yeah, these are not my pictures, but it's like the results that I expect to have, because I didn't have the chance to do the experiment here. So some fluorescent expression here before the PFAS expression. And maybe one last thing that's very important to say here is like, if we have like a PFAS concentration that is over 100 milliliters, that means that it can be like, it can create a toxic environment with for E. coli, and then it kills bacteria, all that makes them like, yeah, and these are like some visuals that I created, or like the way that I would like to have. So some future work is that based on the folding of the protein, we could have like different ways of fluorescent expression. And then that's all. That's the tool I used. And
All right, Alex, great job. Just one quick question is that, you know, again, and just congratulations again. I know it's been a whole journey for you as well, getting here. So I really appreciate all the work. Can you talk a little bit about just like the sensitivity of the detector? Like, you know, if you want to ultimately use this in buildings, you know, or you know, how sensitive can this biosensor be for detecting PFAS? And sort of like, or like, what is like a toxic level of PFAS, and how is the biosensor sensitive enough to actually detect that?
So based on my data here, so I can start like 0, 0. So I have like this data here. So if we have more than 100 milliliters concentration of PFAS, that means that it can kill E. coli bacteria, or makes them dysfunctional. So I would expect, if I was able to do the experiment, I would see if there's like, there's a way like to test it. And they seem to be very, very sensitive, because I found some literature that they mentioned that if we have like 0.01 concentration of PFAS, so that means that
the biosensor
detects the concentration. Yes. We didn't have like the order never arrived. So
Which order were you looking for?
the plasmid. Yeah, but I was planning to do it after, like, maybe next week.
Okay. All right, Alex. Thank you. Thank you again.
Thank you so much.
Again, I appreciate all of the design students that are doing bio for the very first time. So again, y'all are doing a great job. Sophia, up next. Yeah, it's a great to see all of the biosensor projects, and we've got another one coming up. So, Sophia. Okay. All right, Sophia, the floor is yours.
Hello, I'm a first year at MIT. I'm studying computation and cognition and art and design. And my project, in a couple words, it was a dual plasmid cortisol biosensing system. And so I think I'll start with the background. So the reason why I wanted to make a project like this is I work with mice, and sometimes that could personally be a struggle for me, because I know that oftentimes they're suffering a lot and we can't see it, because as prey animals, they're very good at hiding their distress. Here I included this is a very comprehensive study that looked at over 4,700 different studies, and it documented the different causes of death for laboratory mice. And as we could see, some of them are spontaneous, some of them are conditions that, if detected early on enough, could be treated and preventable, such as dermatitis or ocular wounds. These are things that mice don't necessarily have to die from, but often do because it goes undetected for a long time because they seem to be acting normal. In addition to just the welfare of mice in general, stress levels in mice can also affect their cognitive abilities, and I do neuroscience research with Grintoy knockout mice, which basically they are genetically predispositioned to display symptoms of psychosis, which means that their normal levels of behavior do not match that of control mice, which of course is our standard for what's a normal mouse, what's a healthy mouse. Same with mice that are used in studies for Huntington disease or other neurodegenerative diseases. So the idea behind this cortical biosensor is that it will fluoresce green on a gradient in response to cortisol amounts detected. Here, I'll go over the circuit real quick using these graphics, but basically it's two plasmids. Plasmid A is the sensor, and then plasmid B is the reporter. Plasmid A includes two important regions, the ligand binding domain and the DNA binding domain. So cortisol, or a synthetic analog dexamethasone, bonds to the ligand binding domain of plasmid A. And then the DNA binding, it then creates an activated dimer, an activated GR dimer. And the DNA binding domain is what helps it bind to the DNA on the next plasmid, which involves a GR promoter, which basically is a sequence of just GR, GR, GR that pairs up. And so the more GR that is generated from plasmid A, the more, as you could see, the GR dimer is what leads to the transcription of GFP. So the more that binds to the promoter, the more GFP is transcribed. So this is how the gradient function here works. And so I will now go to the aim. So aim one is to just have this genetic circuit working in E. coli. It's being transformed into E. coli. And this would be assayed using a induction assay where you could measure the fluorescence using the OD600. I'll go into that a little more later on. The second aim is to integrate this into a form that could actually be used. And so what came to mind for me was a patch that goes on the skin. It's non-invasive, so it's much more likely to pass certain bioethical testing standards because it's impermanent. It would be a silk fiber, most likely patch that just goes on the skin here. And then that could be used to monitor stress levels in lab animals, not just mice, but other organisms as well. And then aim three would be to take this and get really creative with it. First of all, it could be a useful self-care tool for humans because I think that a lot of us live in a constant state of elevated cortisol and it becomes our new normal. So we don't necessarily notice all the physical or mental symptoms that living in like chronic high stress can cause. And so kind of having a visual reminder that can help people be proactive about self-care and have a better understanding of what's going on with their body. This is also something that I was thinking about, but it could also be as a form of self-expression. I mean, not to like refer to emo Tumblr, but a lot of people do use stress and anxiety as something that's a big part of their personality. And so I was thinking about like even fashion wearables such as scarves or shirts that have this kind of along the skin, so you could see it reflected in like different amounts across your body, or like compatible like bio-tattooing or permanent or impermanent. And it could be really cool to just kind of have that glow on you if you're a stressed person. So I'll go over the experimental design very briefly, but first I designed the two inserts and inserted them into two plasmids. Then here is a very long overlook of my exact experimental design. Unfortunately, my primers for Gibson assembly did not come in, so I was not able to so much as start. However, I was thinking about what results I would want to see. And so, as I said, my final test would be an assay where I would dose different amounts of DEX. And I have here a diagram showing, so the top row is of course just my two plasmid system. And for 0, you would hope to see 0, then a gradual increase, and then a plateau. This is in reference to some other studies I saw that also use bioluminescent or fluorescent genetic circuits to detect other molecules. And so you would measure the OD600, you would get raw scores that would maybe look something like this. You would normalize them, and then you would get a sigmoid curve, ideally. And so I'm really interested in carrying out this project further. And next steps would, of course, be to refine the circuit. I was also considering using a T7 promoter to also induce with IPTG to see how that would affect the bioluminescence and if they get better control. And then from there, as a design student, I'd also be very interested in exploring the patch design itself. And then after that, the possibilities are endless if you want to think about having a lot of stress that way.
Okay. Sophia, again, I appreciate your, what was your double major again? It was
694B.
694B. Okay, great. Yeah. So art, comp sci, computer science, a really interesting combination. Maybe just and I also really appreciate. I know you weren't able to get your primers in time, but I appreciate your kind of mapping through the different types of data you would expect and the experiments that you run in detail. So this is really nice. Maybe just when you talk a little bit from on the art side, like how would you envision? I don't know if you've got some thoughts on some of the types of.
I do have a graphic here, but this was a project by a lab at UPenn where they did a whole series of kind of bioresponsive materials. And some of the materials they made were sweat-informed pH sensing. They had scarves, and this is after it has been worn. They also have this very interesting shirt, which was a grid of dots across the back, where basically a person would wear it and they go outside and get sweaty. And it was a color gradient, which is pretty, mapped straight to pH. And it was very interesting seeing like the sweat distribution across the backs of the wearers. And so I guess the kind of idea would be to not just monitor a biomarker in a single spot on the body, but to see how it kind of maps across the body to gain understanding about oneself and also just for artistic expression.
But you're looking at cortisol and sweat,
yes.
basically.
Well, I'm not looking at sweat, but this project would be, but cortisol, and then this could also be expanded to other biomarkers. You just have to tweak the circuit slightly.
But sorry, but for you and your interface, what? Because if you want to embed this in the material, like, so it's touching the skin though, right?
Yeah, here. Okay. I could go back up to this very, oh my gosh, sorry, my experimental, this very basic diagram, but you'd have silk fibrin, you'd have the plasma itself, and then like microneedles.
Oh.
Yeah, like
Okay.
Yeah. Got it. So not completely non-invasive, but more likely to be approved for use.
Got it. Okay. All right, great. All right, awesome. So yeah, thank you so much again. Okay, we are getting close. Last 45 minutes. I think we've got Alex and Xiang. So we've got a double. So we're gonna do 12 minutes for you guys together.
Yeah, I'm Zion.
Yeah, why don't you guys grab a seat? Yeah.
I'm a first year at MIT, majoring in sixth floor, which is AI and physics.
I'm Alex, also first year at MIT, majoring in sixth floor. So I can first briefly describe our abstract. So capsaicin is paradoxically clinical viable TRPV1 agonist and a potent irritant. It's like kind of what makes food spicy. So it's known that native bovine casein cells, which are basically like protein complexes from milk, can solubilize capsaicin. So we hypothesize that like we could re-engineer casein with like modern ligand-aware delearning tools, kind of like increase capsaicin binding, and then basically translate it, manufacture it, purify it, and test finding to see whether like our design actually improved it.
So I can talk about a little bit of background. Why capsaicin and casein are important. So casein is a milk protein. Casein is found in bovine milk, and many of us know that it can help mitigate spiciness. But capsaicin is not just about spiciness. Capsaicin has three main uses, or three main important applications right now. First, it is actually very important for chronic pain. It helps. It helps like for pruritus and arthritic pain, and also in recent situations, since police use pepper sprays, that can lead to like a huge proliferation of pepper sprays, and this can be very dangerous for communities. And the casein, a high affinity casein, can help mitigate both of these. It can enable slow release capsaicin patches, because current patches can lead to a significant irritation and will well, that's not good. You don't want a product that just hurts your patients, and can also allow like protesters to like practice their rights. And the third thing is there's like been some investigation into like therapeutical aspects of capsaicin in the human body. And of course, slow release is very important for that. So current casein doesn't have as much high like binding affinity. The KD is, I'm not sure if you have it here, but it's about 45 micromolars. The lower the KD, the better the binding is. So we're aiming for a KD of less than 0.5 micromolars through our engineering process. But this is a very high technical challenge. Current methods are mainly focused on protein-to-protein interactions, such as AlphaFold, but we are engineering for binding to a ligand. And that was quite hard.
Yeah, so I can talk a bit about our kind of like vision and impact for this project. So like kind of echoing what Zion said earlier, imagine a fast-acting molecular sponge I can spray on the skin, or like, use to like wash anything like has like a huge amount of capsaicin contact, or just embed behind that adhesive is 8% capsaicin patch for like this arthritis applications. So if we succeed, these three arenas are kind of like what you see. So we have neuropathic pain clinics, which is like the 8% Q10 capsaicin patch, law enforcement, occupational safety, show variable efficacy in OC spray. So, we could improve that. And then also people who generally see capsaicin's metabolic benefits, because these have also been proven, like not super rigidly, like rigorously, but microencapsulating capsaicin with like improved casein for better controlled gastric release. So basically, like the things that we've done in this project that are special, so like ligand-aware design, as you mentioned earlier, most current programs only allow a limited set of protein-protein interactions. So we basically like, combine a bunch of existing deep learning tools into one pipeline, as well as sequence the function loop is like much shorter. So we had a couple of bioethical considerations as well. Of course, safety, non-molestation sense. We don't want our engineered protein to have bad side effects, especially if it's like being applied to the skin. Military police forces might gain first access with regards to like resistance to pepper spray. And then like a capsaicin eraser might embolden excessive OC spray deployment, kind of like a dual use. So to mitigate these, of course, we can do very rigorous in vitro selectivity screens, try to ensure high compliance on manufacturing, and then basically have good oversight over who has access to these products.
And for the aims for our project, A1, of course, is to design a series of very high efficacy casein variants in silico and then produce those in E. coli in order to evaluate their efficacy in a lab. And aim two, and so that is our current goal. And aim two is to introduce the most promising variants in mammalian cells, such as variant CRISPR. So we want to be able to actually manufacture these in like cows, for example. And that will enable us two of our aim-3s. We have two separate aim-3s that we are looking forward to producing. So first, we mentioned before how we have pharmaceutical applications of slow-release capsaicin patches, and that will require clinical trials and like design of the patches themselves. And so yeah, so that's like a long-term process. We need to prove that our patches actually work and actually don't irritate patients' skins. And we also, of course, need to market and commercialize this product. And the second one is a more commercial approach. So, for example, for like anti-pepper spray, for anti-pepper spray, or when you produce those in cows in like very high quantities.
Okay, so I can talk a bit about computational procedures really quickly. But basically, we take like structural information about like how these proteins are shaped, autodock them, use ligand MPN to generate like a huge bunch of like possible proteins, and then basically screen them, first with T-mol, then autodock again. And then we basically got like a top 20. We added his tags in benchling and order with twist clonal genes in the PET 20 backbone.
So we filtered about 100,000 proteins down to about 16. And so for our computational results, here's what we got. We actually, I think we should look at this diagram right here. So the higher is better here, and the baseline is shown in red. Baseline is shown in red, and as you can see, for both our AlphaFold and folks, the produced proteins have like significantly, well, in this case, like more negative binding affinities, more negative binding energies, which translates to increased binding affinities, which verifies our claim.
Okay, let's just get to the experiment. Sorry.
Oh, okay, we're good.
Oh, what?
Oh, okay. All right. Okay.
Slow down a little. Okay.
All right. So we're gonna talk a little bit more about like how we did this. So, we initially docked using AutoDock GPU to just get an initial like binding site that looking at NPN can then optimize the protein sequence for. And then we look at NPN to generate a very large bunch of sequences, but most of those sequences are kind of trash because it does not take into account the protein structure, so most of them are not valid. So how we mitigate that, we use TMO to analyze the structural stability, and we computed Rosetta energies, and about like it top like actually
Basically,
I think
we took the sequences, like refiltered them.
yeah, we filtered them through TMO first, yeah, and then after that we redock using AutoDock GPU to compute the new binding energy scores, which are the graphs you see here. And these help us verify, like these help us generate the proteins which actually bind to capsaicin the best. Yeah, and then Alex can talk about how we appended his tags and toxical genes, very standard procedures to help us purify the protein.
Yeah. Do we talk about these? Yeah, we talked about these. Okay, so now I can talk a bit about the experimental procedure. Just full screen this a little bigger.
Opening the tab.
This is fine. Yep. So we just got our dry DNA resuspended and introduced. So we prepared kinemycin plates and did our transformation. So here's kind of like a picture of our plate, kind of color colonies here, which is nice. We inoculated liquid culture, induced, and then centrifuged to collect the wet biomass and lysed. Did like basically cell debris purification, and then basically did purify with our histag proteins because we used PET28, so our proteins had like histags on them. We could use nickel resin, and then finally, we got our purified protein. It's kind of blocking, and then we detected it with like Bradford assay and SDS page with chemicity. Basically, just confirmed that the protein that we were purifying is like actually there. Yeah, here's some more pictures. These are the cells from 2016 that we found.
Yeah, all the procedures are in the links. We follow the standard procedures.
Yep, it's all here. And then here's like our Bradford assay results. So the black points are kind of like the things that we collected, like the standard curve. Then we fitted a line, and then we found like some pretty strong, successful purifications with like protein concentration around like 100 micrograms per milliliter. SDS page, we got one bar which is like pretty clear. The other ones are less. There's another faint one here. It's like really hard to see in the photo. And then also on this other gel, we had some more. So these ones you just can't tell in the photo at all.
How many proteins are you trying to?
So we tried to do a total of 17. We ran out 8 on this gel, another one on this gel. So of the 8 on here, like only one really showed up, but we expected all the
Philadelphia size in this? It's about 20. It's actually not 24. It's about 26.
So we measured around like this.
This is between 25 and 37.
Yes, between 25 and 37. We expect
like 27, 28, 26.
Yeah,
because like the his tags and everything, the modifications
added some stuff.
like change the weights a little bit.
Yeah,
so we're not 100% sure what the exact weight is of each of the proteins have variations because we change the sequence. Yeah.
Yeah, but so this bar is like roughly, and there's like another one. I don't know if you can really tell in the photo. There's another one over here.
Yeah, so out of like about nine samples, this is the 9 samples we tested. We didn't have a lot of gels. We only had 2 gels, so we had 3 of them show up on our thing. And from our Bradford assay, we also had 3 of them that are positive. That we can tell that there was a non-zero concentration, like yeah. And then we didn't get to do this part. This is the only part we did not get to do, which is the measurement of the ITC. ITC helps us verify that our protein has a high binding affinity to capsaicin. And that requires some special equipment, which we will get access to, I think, this week. Hopefully this week. So this is like a theoretical.
So discussion in future work. So we noticed that Bolt's protein structure had better binding charges because Bolt's is kind of like more aware. It's able to basically like fold the protein, being aware of what the ligand is instead of off fold, which just takes the protein. So transformation was well validated. Our SDS page result was successful, giving us the band that we expected. In future work, we would like to do like a larger liquid culture so that we could collect more protein and have like a higher concentration and do ITC, because like with our current samples, like ITC is like not really feasible. And then that's basically it. Yeah.
Yeah, great job, guys. Glad you were able to get so far with all the protein work as well. Questions? I guess I have one question when you go back to your computational approach. So you had you said you were exploring like a couple 100,000 and then you narrowed it down to 17.
Yep.
So just can you say again, like what were the 17, like what were the properties of the 17? Like how are they, why those 17?
So those 17 were sampled by our final step using Autodoc GPU. So those had the lowest binary energy between capsaicin and our protein that we computed. So 8 of those were from AlphaFold, 8 of those were from boats, and then 1 was the control casing that we used.
Got it. Okay. And then so using the ITC is ultimately like the assay you're going to use to actually test
Develop
this. Okay. Yeah. And do you have anything about just like for those 17, like where computationally, like do you expect them to express, like do you fold well? Is that part of your selection process to get down to 17?
Yeah, so like the folding we verified with Timo. So we expected them to like fold with like the same level of stability as casing.
Yeah. So you do see like a little bit of like stuff here. So when you see 2 humps, I mean, that usually means something is wrong. And this hump right here, for AlphaFold, that means the protein doesn't really fold well. But as you can see, all the boats proteins fold well. They have a very low like there's a conformational energy as well in the binary. So, yeah.
Great.
Go ahead and place in the next version of Pulse. So Pulse 1S.
Oh, well, we don't know.
Okay, there you go. All right, guys. Well, great job. Great thing. Awesome to see the computational work and actually translate full on into design and testing in the lab. Really, really awesome. All right, Cyrus, Smello World. And all 10 hours tomorrow.
Yeah.
We are going to set up a special badge for those of you that like participate in the full marathon. We actually are going to keep track of that. So anybody that does, there's going to be a special award waiting for you. So thank you all for getting through this first 2.5 hours. And Cyrus, over to you.
Oh, thanks. Hey everyone, I'm Cyrus Clark. I'm a research assistant in the Tangible Media Group at the MIT Media Lab. And my project is called Smello World. So basically what I research at a very high level is the effects of generative media, particularly on the planet. And I'm very interested in the tangible effect it has, particularly with things like carbon dioxide, energy consumption, and waste. And that's why I'm very positive about and very optimistic about DNA data storage as a potential solution to some of the problems that these technologies create. But there's a number of bottlenecks with the technology that currently exist. And those things are not going away very soon, particularly around the cost and the amount of time that it takes to sequence the data that's stored in that medium. But at the same time, there are new advances that are occurring, particularly with things in this region here, which I find really compelling, with diagnostic tools which are used for analyzing data, where we can basically see very quickly information stored in molecular formats. But those tools are very diagnostic, very analytical, a little bit cold and dry. And I'm very much more focused on expressive and consumer-facing types of applications. So I'm interested in this region here, where we can reapply these technologies in a more expressive format. And so the innovation in my project is really all about combining different parts of technology, whether it's DNA data storage, synthetic biology, or using things like programmable logic with total switches. And so that's what I've been doing in my design work. And so I'm going to focus mostly on the design, as I haven't got to the experimentation part yet, but I think you'll see why in a moment. So the aims of my project, the first aim is to create basically the hello world of this kind of programmable system with DNA data storage. And that releases a scent as the output. So that's why I'm calling this project Smello World. And I'm designing an experiment basically with plasmids and inserts that will be using a toehold switch as a way to control downstream expression of a scent gene, ATF1. That's very difficult to quantify. So I'm using also GFP as a way to quantify the output. So I can also have a different way of understanding the results. The secondary aim's a bit more lofty. I've been speaking with Peter at the VS Lab, and I'm interested in basically using cell-free systems to also do something very, very similar. So I can have more portable, biosafe versions of these biosensors and these types of applications. But from speaking with him, it seems very much out of scope in the time. So that's something longer term as part of my research. And then the third aim is to create a generalizable version of this, where any kind of multimodal logic-based biosensor could exist in terms of input and output. So my experimental design at a very high level looks something like this. There's a trigger sequence based on data stored in DNA, which is the Smello World text file that I've created.
That then is read by a toggle switch. And then if the toggle switch is open and on, let's say, we can have the center expressed. So the overall design is kind of here. We've got the smeller well sequence, a toggle switch, reporter genes, and a backbone, and then that will all be basically cloned and assembled with the Gibson assembly and transformed into E. coli. So let's get into the actual design parts. So my backbone is PUC19, cut within the MCS region of LAC-Z for blue-white screening. I selected BAM-H1 because that sequence is not present in any of my other parts, so that makes it very convenient to use. For my insert 1, the main part is, of course, the sequence itself, the smeller world. So to actually get that into DNA format, I found a new paper, which is quite promising, with a new codec to take a text file or any kind of information file and encode that into DNA. So that's using something called Wukong codec. And basically it takes the text file, creates a sequence of DNA. And then I created an insert here, which has other elements like an RBS, a promoter, and also quite interestingly, this double terminator. Because when I was designing this, I noticed that my backbone and the insert 1 both had the LAC promoter, and we want transcription to only occur in my part 1. So the double terminator kind of creates a stop between those two to make sure that that works as anticipated. Then the toll hole switches, which are quite interesting to work with. That's the first time I've worked with them. The way they work basically is that we have this unpaired region here, and then a toll hold structure here. In that toll hold structure, in that hairpin structure, sorry, we have the RBS and the start codons, and they're basically hidden away. So we can't get the expression downstream. We prevent expression downstream, and so the gene stays off. But when we introduce our trigger sequence, so in my case, smeller world, it binds in this region here of the toe hold, and then this unfolds the hairpin, and that's how we get this kind of logic gate system, which allows our gene, and in my case, that's Ascent, to turn on. So I had a lot of issues finding good tools for this, and maybe I should just try to do it myself. But eventually I found something that seems to work, which is called toe holder. We use a lot of different elements. The most important things are the trigger sequence, the reporter sequence, and then a few other variables. These are all well documented here. That gives us a lot of results, basically. And then I had to do a lot of filtering to try to understand which are the best results. Here are some of the variables that I was screening. The most important ones are the non-paired count and the absence of stop codons. So basically, these are the results I got. And I basically found these two to be the most promising candidates. I then repeated that for ATF1 and for GFP. So lots of work went into that to get them right, and a lot of trial and error. And then also in terms of the reporter genes, of course, it's ATF1 minus the reporter sequence that's in my toll switches, which I only caught very recently from reading some of the literature. And then in terms of the final design, then you have all of these parts, the encoded message, the toll switches, 4 variants of those. You've got several reporter genes and then a lot of primers. And then we get into the final plasmids, where 4 plasmids. And in terms of the experimental results I'm anticipating to see, from the literature, it seems that the banana scent that is produced should be human sensible. So I should be able to smell this. So that's going to be a very qualitative output. I also have a quantitative output with my GFP, which should look something like this based on the toll hole switch is functioning or not. So where the toll hole switch is on, when the toll switch is basically blocking downstream expression, I shouldn't see very little very little fluorescence. And when the toll switch is opened, we should see obviously normal levels comparable to positive control. And so, yeah, right now I'm pretty much ready to start my experimental work. I think the order should be coming in soon, and I'm quite excited to just get my hands on that in the lab. And then from there, I want to get pretty quickly onto designing this within a cell-free system. There are some issues with doing this, which Peter has already spoken to me about. It seems feasible, but there are some quite difficult issues around the fact that, of course, if I'm working with scent in particular and alcohol as a substrate, there's an issue with that destabilizing the cell-free systems that they currently work with. So that's probably the major bottleneck that I'll be facing with that. But there are probably ways around this. So yeah, that's all. And welcome any questions.
Great. Cyrus, yeah, I guess just maybe since also you're a Media Lab student, maybe you can comment. So is this, how much is this just project folding into your overall kind of research for your thesis? Is there?
Oh, it's basically my thesis. This is like the precursor this is like the building block, let's say, of my thesis. My thesis
Great.
is a bit broader, a bit bigger in terms of thinking. So it's around this thing called regenerative media. And this is one block of it where I'm thinking about how we can interface with data stored in DNA. And this is a nice way to experiment with something at a kind of like fast level with E. coli, but it's, as you see in my previous work, it
Yeah.
can't get out of the lab.
Yeah.
And
yeah.
people want to see and touch this, especially with something like scent, they want to experience it. And that's why I'm quite excited about working with Self-Read to try and develop that into like biosafe sensors that could be deployed.
Well, we look forward to wandering into Hiroshi's lab and smelling the lab.
Yeah, right.
Oh yeah, Michelle, good.
Yeah, what do you envision for the interaction between, I guess, the sensor, also the active stimulus and humans, and then like, where do you go?
I think, to be honest, I think because I've got a background in design. So typically the way I've been working is having these really lofty visionary goals of those kinds of things, those types of interactions. And what I'm trying to do now is just like focus on what the experiments offer me, what the science really offers me, and then work with that in kind of reverse way. So whereas typically I would have these very lofty goals of, oh, I touch a plant and it releases a scent, and I interact with the other organism, it seems very like non-feasible based on what I can see from the research I've been doing. So I'm trying to work in that kind of manner now.
Awesome, Cyrus. Thank you so much. Great job.
Yeah.
All right, home stretch. Last couple presentations. Alex, come on up. So we've got Alex, Vicky, and Vivek. Vincenz is not here, right? So he is going to join on Zoom. All right, we are running out of time. So just say I did not know. Let's do Alex. Vicky, if it's okay, I'm going to have you go last. Okay. All right, Alex, go ahead.
All right, so antibiotic resistance is one of the largest issues facing medicine in the future. It's like the third leading cause of death in most countries, or third world countries right now. And it can be caused by a lot of things, just like people not taking their antibiotics properly, over prescription of antibiotics by doctors, a lot of that. So there's a lot of reasons why we need to solve it. Because a lot of people can die from it, and this leads us to my project, which is antibiotic resistance. And so there's a class of antibiotics called beta-lactams. These include some very common ones such as penicillin and ampicillin. And these are, you're gonna get the nice little picture here. So with these beta-lactam antibiotics, when you take them, they bind to the penicillin protein. And then once they bind together, they can destroy your cell, and that kills the cell. But cells that are resistant to these antibiotics produce beta-lactamase, this little green guy. And that hydrolyzes the little ring in the beta-lactams, so it destroys it. But we can produce our own protein called the beta-lactamase inhibitor that binds to the beta-lactamase and destroys that, so it can't destroy the blue thing, which is the antibiotic. And then it can let that destroy your cell. And so we have this synthetic protein called TEM 3.0, and that protein creates very specific conditions with, called exploits like APRs, which is very specific conditions where the beta-lactams, the beta-lactamase, the antibiotic resistance, under those conditions, the proteins will misfold. And so we have the amino acid sequence for that, and we can reverse translate that amino acid sequence to an RNA sequence, then reverse transcribe that into DNA sequence and make that into a gene and codon, optimize it for E. coli and everything, and then put it into a plasmid, have the plasmid produce that protein and make it ampicillin unresistant again. And so the vision for this is, right now, we just want to prove that we can, since this is a synthetic protein, I don't know if the paper produce it from a gene or something, or they produce them other way. And so I want to prove that we can produce it with a gene and that it would do the thing it's supposed to do. And so first of all, it's going to, our first aim is to kind of prove that in the lab. The second one is to find a way to deliver this with the antibiotics. And then the third way is to, so this kind of doesn't hit the root of the problem because it's just a gene that like is the resistance. What if you just like take care of that gene? So the third way is to like do something with gene drives and take care of antibiotic resistance at its root. And so our bioethical considerations are, we don't know if this protein is safe for humans or not. And we don't know like what its effect on living things are going to be. And that's like the main one. So we're going to go to the experimental planet and timeline. So we have this right here is the amino acid sequence of 10.0, the protein, and then we can reverse translate, reverse transcribe, and then once we codon optimize for E. coli, we get this. And then using the DNA design construct, we combine that 10.3.0 with GFP to see if we produce protein or not, and kind of put it into like the whole like T7 promoter, RBS, start, vector overlap, all that, and then we get this using PUC18. So initially, we wanted to use it with PET28 because since we're trying to turn off ampicillin resistant, there's no way to see if it took up the plasmid or not if we're using ampicillin as our antibiotic. So we need to use a different one, which PET28 uses kanamycin. But we couldn't get PET28, so we're using PUC18, and that one only has ampicillin resistance. That's fine. We'll get that into that in the discussion real quick. So we resuspended the gene fragment. We determined the concentration of the gene in the vector. We restriction digested it using EcoRI and HIME3. And then, so we only have 0.02 nanograms of our PUC18 plasmid. Anywhere from there to 0.1 nanograms. So the gels aren't going to look great, but we ligated that into a plasmid. We transformed it into E. coli, and we transformed it into DH5 and BL21, and then we plated everything. And this is our gel. So our first gel from the restriction digest, we expected something from 2,400 base pairs right here. And then we expect another thing at 800 right here. And the rest of these lanes are our PET28 PCRs. That didn't work. But we do have our plasmid. And then from this one, after the ligation, you can just barely see something right here, which is where we want it. Just keep in mind, we have like almost no nanograms of our thing. But once we plated it yesterday, we checked it this morning, it worked kind of. So ampicillin resistant, everything grew as it's supposed to grow. Non-ampicillin resistant only grew up until 1 milligram or microgram per milliliter, right? BL21 has our ampicillin resistant gene, and the thing to turn off, it has ampicillin resistance. It didn't get turned off. So that didn't work. But DH5, so either two things happened here. It didn't take up the plasmid at all, and it just died. Or it took up the plasmid and expressed the protein, and that worked, and it died at 0 or 1 microgram per milliliter, like it's supposed to if everything worked. However, we have GFP in our thing, and nothing is green for these top two. So we might have to lean towards it not working. But we'll have to double check that and see. And so our results have very mixed, yeah. And we need to see like if the BL21 did something differently, GH5, we just need more nanograms of plasmid. We had nothing to work with initially. And I also, when I digested it, I picked up the wrong pipette and put in too much of the restriction enzyme. We had to adjust the everything for that. So, but we had no more plasmid, so we kind of had to just work with that. And for future work is to mini prep and Sanger sequence to see if those colonies, the DH5 and BL21, actually took out plasmid and are producing like protein or not, and like do like the whole Kumasi and Western blot.
Alex, we gotta wrap up right there. You're over time. Like, good job. So just I understand again, your overall goal here. So ultimately, how do you imagine this? Because all this work here is still like you kind of producing the protein and testing it. What is the longer term vision? Like, how do you can you say again, how you imagine this being used in the world?
So if we have this plasmid and it works and we make it, so we have this gene and we can do something with gene drives to make it more dominant than antibiotic resistant genes, then nothing should be antibiotic resistant. And the gene drives to do what? To like, what would you propagate? So, gene drives are propagating a gene through a population of something like mosquitoes or something. Like, what's the, through like just your microbiome in your body, right? Like, because bacteria have like horizontal gene transfer, right? So if you just give enough bacteria with this gene to make them unresistant to antibiotics, then at some point everything should get better.
So you want this as a human in a human therapeutic context. Okay.
That's better than taking out the beta lactamase gene.
So if you could just do that, that would be better.
Gene drive can do that too, but
that's the aim.
This might be better. This might be better because if it goes out in the population, it will be antibiotic to those gene drives in the brain.
Yes. Yeah.
One quick thing, DH5 alpha is not an expressing form, so if you have the C influence with GFP, you might not have that.
Oh, okay.
So that may be why you didn't see it. You might not that it didn't work, it might also be that it didn't.
Oh, okay.
Yeah, because you might have induced it, but even DH5 alpha won't do that very much. So.
Okay. All right, Alex, thank you.
So in our last 12 minutes, Vivek, is Vivek here? Okay, Vivek, come on up. And then can you check if Vincenz is online?
Yeah.
Okay. All right, Vivek, go for it. Oh, this is let's link from the sorry. Oh, sorry. Here. Okay. And everyone, I apologize. We may go a few minutes over in class today, so I hope folks are able to hang out for 12 minutes after.
All right, hi, I'm Vivek. I'm a sophomore in 601 electrical engineering at MIT, and I am working on a heat stroke tattoo biosensor. So when the human body overheats, it's not always obvious. You might feel a little off, but inside things can go south pretty fast, especially when the core temperature hits around 40 degrees Celsius, which is like the danger zone for heat stroke. So I'm hopefully working on a living tattoo that changes color when the temperature exceeds this heat stroke threshold. And so this system would use the B. spores. These are like dormant survival capsules that would sit safely in the ink. And they'll be engineered with a heat-sensitive promoter called GROE and a pigment gene called amylcp that would make the purple color right here. And when the skin heats up past about 40 degrees, the spores should wake up and then start producing the color. So far, the design is fully digital. I use Benchtone to model the DNA, and I plan to build this with Gibson assembly. And then once built, I'll insert the plasmid into acetylus, and the resulting spores will be dried into powder for the tattoo. So some quick background, why is this important? Heat stroke can cause brain and organ damage, but symptoms can be vague, like lightheadedness or nausea. There are some wearables like watches or sensors that rely on electronics, but these can fail silently and people might not always want to wear these. And then at the same time, synthetic biology, however, is beginning to explore these living materials like biosensors to respond to the environment without these electronics. But so far, these spore-based biosensors have only focused on like toxins or nutrients. I found this one article from MIT that did something like this. So this project should hopefully fill this gap by showing how this spore ink can give a visible warning when the skin overheats and then fade back, hopefully once it cools. And so why the spores from this Bacillus bacteria specifically? This is because these can survive for a long time while dry and they'll just stay inactive, but they should wake up when the conditions are right. And then the bacteria should read the DNA program and do whatever we told it to, in this case, make the pigment. And then since the spores are also dry and stable, they could just hopefully be simply blended into the tattoo powder. Skip this. So I'll talk about some bioethical considerations on, I guess, some potential issues. It could be like false alerts or no alerts at all. Spores washing off, genes jumping onto other microbes like in the environment, so like contamination, and also interfering with the skin bacteria itself, so messing with our microbiome. So some future directions, I guess, could be to include some sort of kill switch or maybe some peel off format. For project aims, so aim 1 would be to build a circuit, I guess using benchling to design the grow-eat to male CP loop, and then order and amplify the parts, of course, and then fuse them with Gibson assembly. And then the goal for aim 1 would be to create the working plasmid that activates the color with the heat. Aim 2 would be to insert the plasmid into the bacteria, let it form the spores, and then dry it into the powder, and then test this on like a gelatin pad. And then aim 3, this is more of like a future directions. I guess like build other switches, not only for heat, but for UV and light, maybe pH as well, and then like mix these into different tattoo inks to create like the multi-color stencil that would warn of multiple dangers. And then I'll quickly summarize experimental design. Step 1 is the digital stuff, benchling, creating the, having the PHTO1 backbone, inserting the grow-eat promoter, the male CP pigment gene, and then the overlapping ends for Gibson assembly. Step 2 would be the PCR and the gel check. And then afterwards, Gibson assembly. Step 4 would be to transform and screen these into the Bacillus. Step 5 would be to create the spores and then dry using some media that triggers the sporulation. And then I'll mix these into the tattoo inks and then freeze dry them to make the powder. And then step 5 would be the testing. I'll apply the powder to the gelatin skin like I mentioned. And then also I'm considering a biosafety check to make sure they don't spread. I'll first like spread them on a plate. And then if nothing grows, this confirms that the spores would stay locked in the powder. These are our steps. And then since I haven't been able to start the lab work yet, think ahead of what the data would look like. I sketched out 2 ways the tattoo might behave based on how treat might trigger the system. The first possibility is like a gradual response, where the color intensity slowly rises with temperature. The second is more like a step function, no color at all until you hit the threshold, and then a sharper switch to the color. And I thought about this. I feel like from a user safety perspective, the step-like version is better because it avoids confusing someone with a sort of like halfway color tattoo, which might not be as clear. And then I'll talk about a discussion a bit. So for color, this gene has been shown to produce the visible tint, although it would take a little longer than ideal. And this is a normal bacteria like E. coli. But because the grow-eat promoter turns on faster, I'm hoping that this would cause the color to appear and soon enough to be able to serve as a good biosensor for heat stroke and not have it be too late. And then for future work, like I mentioned before, I'm considering the multicolor warning system. And then another idea in case the color does not go back is to like add a degradation tag so that the spores self-destruct. Yeah, that's it.
Okay. Thank you. Another nice biosensor one. Questions, comments from, oh, yeah, let's say questions, comments for folks? Questions? I guess one question for you is, so what's the first experiment you're gonna work on? I know you weren't able to get going in the lab this time, but what's the first thing you're gonna be testing?
This first thing I'll do the basic reference of this assembly.
For working with your to express the protein?
Yeah.
Okay. All right, well, why don't we call it there? So Vivek, thank you so much again. Appreciate you. And all right, so everybody, just like if you all can just hang just a couple of minutes after class again, I thank you all so much. So let me stop sharing. So we have one more presentation remote, and then I'm going to do just 5 minutes of wrap up. And thank you, Jimmy, for being here to help take a good picture for us. So Vincent, go ahead. The
All
floor is
right.
yours.
Thank you very much. I hope you can hear me. The irony is
hear
not
you.
lost on me that I'm presenting something about remote lab space remotely, but my health kind of has come in the way of being there with you. So my project is on kind of bringing synthetic biology education to places where synthetic biology usually isn't possible, through the help of kind of remote lab space, right? And I guess what guided me through all of this, and I guess also my experience in education, is tell me something and I will forget it, teach me and I may remember, but if you involve me, I can really learn something very well. And I guess the background information and kind of where all of this comes from is we've all seen in this class kind of the amazing potential of synthetic biology. We've seen ways to cure cancer, fight sickle cell disease, some of the great presentations now, and kind of seeing stress levels, seeing if we're overheating, and all these kind of amazing things. But at the same time, if we look out in the world, there's a ton of people that just like turn away from science and are like, I'm no longer trusting very basic medical procedures, very basic medication. I was kind of shocked to read a couple of weeks ago that there's something called measles parties in Texas. And I was kind of like, okay, there's something that's not working. And I kind of looked deeper into this. And one of the things that, I found one medical talk to talk about it is kind of this distrust is often rooted in a lack of understanding how all of this science works. And for us, that's probably like really strange because, we're surrounded by great labs, we're surrounded by great researchers and everything. But we have to remember that that's kind of really not standard. And so my idea was kind of like, how can somebody that is kind of in an under-resourced school, be it in rural Oklahoma, be it in an inner city in maybe New Orleans, how can they have access and kind of not just hear about this, but also kind of themselves participate if they don't have lab space in their school or in their communities? And I was really excited by, I think, a second or third class on the Opentrons, where we kind of programmed something and then kind of fully automatically ran those kind of lab protocols. And so my idea was kind of, can we figure out a way for people to create lab protocols to walk through all these steps and then have it run somewhere that's kind of maybe not in their community, but they still get the results and they still get to be kind of the people responsible and driving those new things and kind of see themselves as the people. And so the idea was taking a specific lab protocol and kind of automatically scripting it to run on an Opentron machine. And the main goal in kind of education and kind of bringing this is that on the one hand, people are able to understand what's going on, that they learn this. And even if they don't pursue a future career in this, they have a much better understanding of, what is synthetic biology and where does it come from, but maybe also kind of inspire more people from kind of unconventional backgrounds to go into this field and are like, hey, this sounds cool. What other options are there? And then maybe find out about things like iGEM and kind of educational opportunities to kind of look at and further pursue this. So kind of how can we do this? And the idea was, in a technical sense, take an existing lab protocol and then transform it with machine learning, large language models into an Opentron's code that can be executed. And for this class, obviously, synthetic biology, take an existing protocol that I really enjoyed, and we're going to get in more detail, the Gibson assembly, and kind of see, is this something that we could run actually in Opentrons? There's obviously kind of providing access to a lot of people. There's obviously considerable ethical implication of like, if everybody all of a sudden could use these technologies and can work with them, like what are the implications there? And I think one important aspect is similarly to this class, like we've seen in the first lecture, bringing in, making it requirement, like before you use this, what do you think? And kind of what do the people that use the tool then think when implementing this, but also kind of checking on our end or like on whoever is running the lab, is what kind of is the process that's being done and being conducted like sustainable, and like is that dangerous to a certain extent, I guess.
2
And
minutes, Vincent. 2 minutes.
of course.
2 minutes. Yeah, go ahead.
Oh, sorry. Yeah, and on the project aims, I guess the first aim was kind of taking that Gibson assembly lab protocol, kind of because it combines so many things like DNA read and write, and then kind of working with OpenTrons. The second aim would be expanding it to more and other lab protocols. And then in the third step, kind of expanding it and bringing it to those communities and actually rolling it out to them. So kind of quickly walking through the experimental design here. I kind of looked at a way to kind of repurpose the existing Gibson assembly, but kind of bring in something new. I found an interesting protein, TD Katushka, which is a very beautiful bright red protein, and kind of then modified it with twists to kind of fit with our kind of so that we can order it from them. Kind of inserted in benchling. We can see here with the forward and the backward primers as well. Great shout out to the teaching team for helping me verify that, and to Twist for kind of being able to order this protein sequence from them. And then we see here the forward and the backward primer so that we can actually insert it into the overall backbone.
One more minute, Vincent.
Okay. And then we kind of created the lab protocol and started to set up the kind of procedure. Started with initial parts of the kind of protocol and kind of ran through, and we're like, okay, what are the learnings? What didn't work and what did work? And then kind of reiterated step by step by step. And then in the next step, kind of putting this into an instance so that kind of actually users can use it. And kind of 30 seconds, quick results. What we found so far is the scripting functionality, particularly for shorter code blocks, works well. But what's necessary is that there's some specific factors that people need to clarify beforehand, and also some instruction sets. And I guess this is the learning here that should be given with this tool and kind of be in the part of the script to work. Otherwise, it worked really well. So there's some minor calibration issue of like getting the software code to then like perfectly run on the machines. But I think overall, this is like a very promising way to actually conduct like this interesting lab work that we've done and we've had the pleasure to do remotely without having to actually be in the lab or have access to that lab space. Thank you.
Okay. All right. Great job. Just very quickly, just to say one more time for clarity. So, what's the interface? How do you envision this in a learning environment? Like, what would the students be interfacing with?
Yeah,
there?
so the interface, my professor here at the Kennedy School has this open source chatbot interface, and they have like, for them, it's just like a normal chatbot interface. And we would be able to provide a certain set of instructions that really turn kind of normal sentences or whatever and give it specific instructions to work well on an OpenTron, and they would then enter just like, do this and that, or just like a normal lab protocol, like they would. It would then return them a code that they can send to a lab that would, like a node, let's call it, who then would be able to run the code for them and kind of send them back results and be like, this is kind of what your code, the work that you've done has kind of achieved, and kind of the work that you've done, right? And they see something, and it's not just like, oh, I've written a protocol, who knows what it does, but like, here's an actual thing that was built with the protocol that I've worked on.
Okay. All right, Vincent, thank you so much.
Thank you so much.
And everybody, just one big round of applause for everybody for all the awesome presentations. Really, really phenomenal job today. Really, really good. Great job. So just to wrap up, hang with me for just 2 more minutes, everyone. Thank you again. Just give me 1 second here. So just a closing couple of remarks before we wrap up the class. So if I full screen, can you all see this online? Full screen, yeah? Okay. So tomorrow, again, 10 hours of presentations. Anybody that sits through all 3 of these plus another 10 hours, we're going to give you an epic prize of some sort. We're figuring that out, but we are keeping track of this. So I hope you'll join us virtually. And for our global students, again, we're excited to give those of you that complete the course this certificate of completion, and we'll do a little graduation for the global students later on in June. I want to give a huge thank you to this year's sponsors. So Twist, Open Transdermal, Waters, we could not have done this at all without you. All of the support from the reagents, the DNA synthesis, the robots, the mass spec, just huge gratitude and thanks. So can we all give a big round of applause and a huge thank you to the sponsors. Really cannot emphasize this enough. Just all of this experimental work that you've all been able to get done, none of it would be possible without you guys. So just so grateful to all of you. I want to really, I'm almost gonna get a little emotional. I really want to just thank all of the TAs. So, in particular, Suthan, Ronan, Michelle, I know Johannes is here as well, but all of the TAs, I'll start here with the MIT Harvard folks, just so much time and effort gone into this class and supporting you all. I know all of you have been in the lab, especially this past couple of weeks, and huge amount of effort that's gone in, just really thinking about the experimental design, supporting you with all of the projects, the ordering, everything. So can we give a massive, huge round of thanks to all the TAs? Thank you guys. Thank you. Yes. Yeah. Thank you. Again, just so grateful. The class simply would not be possible without you all. To all the global TAs, again, we're going to thank them in more detail tomorrow with the global classroom. But again, huge big crew of global TAs all around the world. So we'll thank you in greater detail tomorrow. And then also, again, huge thanks to all of our nodes this year in New York, San Francisco, Quito, Ecuador, Victoria, Ottawa, Canada, and London, South Korea, and in Tokyo. So you guys will do more thank yous to that whole crew tomorrow. But again, it's been such an epic journey. And again, I really hope you all will come join for the presentations tomorrow. So just to wrap up, I just want to say a couple things about where we are with the future of how to grow. So I'll skip through some of this, but just to say, we're in this process right now of expanding our work with how to grow. We were invited by BioMade to submit this proposal to build out a national network of how to grow almost anything nodes. And so we're really excited to bring on more nodes next year across the country, some of the ones that are shown here in this chart. So very excited about that. We are going to continue to grow the course globally, steadily and slowly. Again, we move at the speed of trust and making sure we've got really good leadership in all these places globally, but very excited for continued growth of the class. And just to mention, I thank those industry folks. We've really grown out the industry council for the class over the past couple of months. So we're bringing on board 21 different companies that are going to be a part of next year's How to Grow. So are very stoked for this whole growth. Yeah, so next year we're going to be able to do even more epic work with all of our new industry council members. So I could not be more thrilled for all of those folks. Some of the members of this council were popping in and out as well. So just grateful for future collaboration with all of you. And I want to say too, and so we've also been building out again another great set of folks to help us think about the scaling of this work into the future. You all have been working on this and touched on this idea of global projects, right? So over the summer, just to say, many of you want to continue doing your work on your projects. So we'll follow up with you in the next month and beyond about continuing and getting some data for your projects, which again, are going to turn into theses and other things as well. So very excited to support that in the coming months. And we also want to support you all in collaborating with us on these global projects. So this phage therapy project is one that you all contributed earlier on to in your protein design work. The neuromorphic circuit project, which we started this year. Again, shout outs to Ron and to Evan. This neuromorphic circuit design project, we also are going to scale globally. So for those of you interested in participating in that, excited to support you there. And I want to close with just sharing a little bit of a story. I shared it with you a little bit with you all, but I visited Ginkgo a couple of weeks ago. And, me and Tom Knight and Jason, we are three handsome guys, but more handsome than us are these robots behind us. So these are what are called racks, reconfigurable automation carts. And so you can put an arbitrary kind of bio tool inside each one of these racks and string them up together to basically make kind of an arbitrary protocol. And so this whole system at Ginkgo is called Nebula. And one of our larger goals is to build out essentially the cloud biolab of the future here at MIT in collaboration with Ginkgo. But in the meantime, what we want to do, Ginkgo has very generously said, hey, we've got so much time and space on Nebula. Let's let the How to Grow community come and play with it and work with it. Okay, so another project we're going to execute over the summer in preparation for How to Grow 2026 is actually working with Nebula and to do cloud lab work and to explore what's possible. And talking with Jason, it was really fun talking with them. They were like, all right, they have all these constraints. We want to have a set of, kind of plates that go in. I don't want to touch the thing during the semester. And we thought, okay, well, we need to figure out what's going to be possible for the students. And so, here at the Media Lab, there's a very famous paper that came out in 1971 by one of the founding faculty, Seymour Papert, 20 things to do with a computer. This is back before people had any idea that you could do learning and other kind of creative applications with computers like we see all the day today. And so what we're going to work on over the summer is this idea of 20 things to do with the cloud lab. Okay, so if anybody that's here for the summer wants to hang out and work with us, we're going to be collaborating really closely with Ginkgo on all of this. So everybody, it has been a super epic semester. I'm going to give it over to Joe actually, and to George, if George is here, to give a final word before we wrap up. But it's been such an honor to work with you all. Very excited about the future of the course and where we're all headed here. So let me see. Oh, George is here. Great. So George and Joe, you guys, I'll turn it over to you guys for the final word.
Yeah, the hour is late. I just want to give a tremendous round of applause to David. I think this course is on an exponential up curve, and it's just really exciting. So,
Final word, George.
Same for me. Thank you very much, David. Doing a lot of heavy lifting all the way through, and thanks to all the students for these remarkably creative projects. And boy, really looking forward to that expanded cloud lab next year.
Amazing. All right, everyone, why don't we come up and take a picture together and we'll wrap up. And thank you, Jimmy, for hanging out. So I'm gonna put this in the back. You guys, for those of you virtually, please turn your videos on so we can see your faces. And yeah, Joe, feel free to come on up over here. Joe, actually, if you have a seat, we can have a couple sit in the front. Ronan and Subin, you guys want to come and sit down too? Yeah, have a seat. Joe, yeah, come on over. Ronan and Subin and Michelle, have a seat. Come on, get over. Yeah, nice. Yeah, you guys.
2 rows, I think.
Yeah, two rows will be good.
Tell people you know you are.
And Jimmy, I'm going to give you another camera to work with here too, if that's okay. I'm going to give you a.
We can, it's at the top. We have to maybe move students to a little bit. Yeah.
Yes. So actually, come sit. So sit down. Ronan, Subin, come and sit down. We need to do a couple folks to sit down. Yeah, come, Michelle. You can grab this chair over here as well. Yeah, come sit. Yeah. Come sit. Yeah, come sit. Great. Okay, awesome. All right, everyone. Let's say grow on 3. Grow. Yeah, here we go. 1,
2, 1.
Grow. Nice. One more. Awesome. Thank you so much, Jimmy. All right, here we go.
All right. 3, 2, 1.
Grow. Great. Thank you guys. Thank you guys so much. And 6 o'clock for dinner at Milan. Just show up. Just show up. I know a lot of you didn't RSVP, but just come. There's going to be good food. So see you all there. Yeah. Yeah. Sure.
Wait, no, yeah. Go. Yeah.